quit, and I'm not sure why, but let's just try again.
Okay.
So far, so good?
Yes, we can see.
Yeah,
Yes.
just want to let it stabilize, but
please go ahead.
okay, I'll start again. So anyway, I would love to give you a flavor of some different approaches we can take with a structural mass spec toolbox to study antibodies and different antibody formats. So if I can advance my slide, we'll get there in the end. I'm not sure what's going on. Okay, here we are. Yeah, so antibodies, of course, we know their basic architecture is shared between different types of antibodies, but there's still different important differences. For example, we have different types of IgGs, if you want to focus on these for the moment, and they have different numbers of disulfide bonds, and also the CDR loops, where, of course, in the variable fragments, where the actual recognition happens with a target, it's quite important how accessible this is and what structural dynamics the antibody has in that particular part. So there are a range of course, biochemical, but probably also biophysical questions which mass spectrometry can help to address. Right, so some of the techniques I would like to focus on today, so I'm not trying to make this comprehensive, are the native and ion mobility methods, but also native and denaturing top-down ETD, quite briefly, because I think for proper top-down sequencing, you will have a talk later from Moritz Den Boer. I will talk about HDX, but I guess particularly emphasize some approaches we take with FPOP to understand antibodies. So most of this is, of course, known to you, but we can do top-down and bottom-up approaches, and there's always a place for both of these, depending on what your question is. And if you're talking top-down, then the majority of experiments are done denatured, but you can actually also do native top-down. And why would that be interesting is something I will briefly mention later, so not say it yet. Bottom-up, of course, is important because we need to verify the sequence of an antibody, but there are also many small changes, not just disulfides, also deamidations, glycosylation patterns, which are done in classic proteomic workflow. That's not something I will talk about today. So the native methods, just very, very briefly, because again, most of you will know them. They are about keeping the native higher order structure of a whole protein antibody or a complex intact inside the mass spectrometer. So you can get information on stoichiometry, architecture of complexes, but also the global size and shape. You can directly detect interactions because they're preserved in the gas phase. You get mass and composition as well. And actually it can be useful because antibodies are above 100 kilodalton. So even if you just want an accurate mass, it can be useful to do that in native mode, because many LC-MS setups for denaturing MS might not work that well when the proteins become too big. But more importantly, when we use ion mobility, we can get some idea of size, shape, conformational dynamics, and various other aspects which might not apply here to antibodies, but if we have interactions we want to study, and we can look at these complexes and study their structure, their stability. Okay. Electrospirionization, as I said, relies on, in this case, when we want to do it native, on using solvent conditions and instrument conditions which preserve folding and interactions. And that's something we can do quite routinely now on different types of instruments. Right, okay, let's start with the actual story with antibodies. So this goes back a couple of years when Yuri Zibin was leading a consortium to compare different approaches which were left to each lab to do top-down sequencing of monoclonal antibodies and constructs, which he was happy to share with the community. And I'm telling that story because it was actually quite funny when my student finally sent in her data, and then I met Yuri soon after. He joked and said, "You've got by far the worst data of anyone who's sent anything back." And I guess we knew that and we anticipated that because we were the only ones who did native top-down. We said straight away, okay, we do top-down, but we are not so interested at that stage in just sequencing, but we want to actually see if we can learn something about the structure. And that's what I want to briefly emphasize. And in fact, also illustrate with an example. So for native top-down, of course, you could still, if you want, from the native state, you can still use collision-induced dissociation. And in a recent paper by Zhou Lu in JAX, he's also shown, and we've seen that before as well, that you can sometimes get interesting aspects of a higher order structure of a folding architecture of a protein from CID. But CID, collision-induced fragmentation, gives you B and Y ions, and as you all know, it is more or less like slow heating. So you actually really assume it breaks the weakest bonds, and that's typically the peptide bond. So you're not expecting to learn a lot of detail about how a protein is folded or what's dynamic, what's exposed in a structure. So a prototypical technique, electron transfer dissociation, and of course also ECD and UVPD in principle belong in that family, is quite different because it uses electrons or radical anions, which then transfer electrons. And these are, you could say, ending up injected into the protein structure. But we think they actually end up on the surface of a protein. So the fragmentation we observe is generally always on the surface. And most likely it even has to do with the position of the positive charges on a protein, where the electron most likely reacts. And the reaction is very, very rapid, so that it's not a thermalized process, and you end up cutting quite selectively the C alpha and H bond in a protein, and that means you make E and Z ions, but it also means that you're not really touching the side chains all that much. And that's interesting for PTMs, which might be unstable, but I guess it's even more interesting when you have native structures or non-covalent interactions, which you then actually preserve, even though you're cutting the backbone of a protein. So, and we were ambitious enough to do that on whole antibodies at the time, and we did that also at the time, a couple of years ago, on a Synapt QTOF instrument from Waters, where we used the, don't need to go through all the detail, but we used the ETD capability. In fact, we developed that a little bit together with Waters, where you have in the source a flow discharge, where you can then make the radical anions from dicyanobenzen, and these radical anions basically travel down the same path as your positive analyte. But in the trap, so you have to switch the polarity, so you have a certain cycling going on with the time, how much time you dedicate to positive ions and negative ions, but then you trap them together in this trap, which is prior to ion mobility. And depending on how you control the gas pressure, and you have to use helium and the wave height, you can actually either keep them more or less separate because the wave separates positive and negative ions, or if you lower the wave height, you get them to react. So it's not fair to say the most efficient way to fragment, but it gives beautiful spectra, and it is a very, very gentle process. We are doing that now as well on a new Eclipse platform, but the data I'm showing here is now still from ETD, from a Synapt. And I'm not showing much data, I just want to show a contrast. So this is actually denatured ETD data from uricibin, and you can see that you can get reasonable sequence coverage. I think we'll have better examples later for Moritz. But in comparison, if you do native top-down ETD, you can actually see how it really predominantly hits the most accessible, the most exposed parts of a protein structure. And even though, coming back to the beginning of the story, even though we had basically terrible sequence coverage, that pretty much relied on what part of the sequence was actually exposed on the surface. And for that, actually, it was fairly good. So what I'm trying to say here is that just doing this here is a proof of concept, but if you have related antibody formats, or if you want to even maybe map an epitope, and you then compare two settings, actually think that native top-down sequencing with ETD, ECD, and if you do it in a certain way, UVPD, can be quite useful. And it's a method, I think, which we will see, I would predict in the next couple of years being used quite a bit more in this context. But it is not something we do routinely. There are other methods most people would want to use, HDX, of course, for epitope mapping. But actually, I think we'll start to learn that this might well be very compatible with that workflow. So we like to call that then conformation-sensitive fragmentation. Now I move on a little bit, and this is work of a current PhD student of mine, Melissa Coxhead, and she works with AstraZeneca, and they have previously, actually with Alison Ashcroft at the time, worked on some formats of an antibody where we start to really ask questions about engineering a structure, changing something, changing a biophysical properties, and then asking how far mass spec methods can reflect or can help us understand what's going on. So here's a long list on the left-hand side with mutations, maybe not so interesting, but if I try to summarize it, this is an IgG1 format, and basically it had to do here with changing the dynamics around the hinge. I'm sure you're all aware how antibodies have really interesting and highly flexible dynamics around the hinge, although it does depend on the antibody class. And here there was some engineering done in the TM position. Actually, the residue is TM, but let's say in this range, and YTE. So there are two sites where some mutations were done, basically one set, another set, or both together. And that was already published at the time, a couple of years ago, that these mutations lead to a decrease in thermal stability in the CH2 domain, but they allow us to actually change the dynamics around the hinge to basically make it more rigid or make it more flexible. We see how that translates into the overall conformational dynamics, into the thermal stability, which is important when you make a product, but it also plays a role in how easily basically the CDR loops, which is where the interaction happens, how easily they can engage with their target. So that this is actually allosterically linked, and it's quite, I would say, still poorly understood how this translates to the whole antibody. While of course we do understand quite well what happens locally, for example, in the CDR regions. So using these different formats, and here we have the full-length antibody, and this is now the wild type, we can of course, here in the top is the wild type, we can of course just use ion mobility, and we can say, do we already see a global change, a difference? Is maybe one more stretched out and one more collapsed, but we all know that we do get some sort of self-solvation of an antibody, and we can call it a compaction in the gas phase. So maybe we are not very sensitive here because they are still able to visit different states, but this equilibrium will be shifted when we take the various mutants and compare them to the wild type. So if we just do ion mobility of the whole antibody, we do not really get that much. Now, there is an enzymatic toolbox as well, and this is quite important when we work with antibodies, and was also part of the study with uricibin. In this case, I've just highlighted two very popular enzymes, and company Genovis also develops these funny names and cartoons for them. But they're basically enzymes which cut above the hinge or below the hinge, and there's some other ones as well. And you can then make very bespoke parts of an antibody, and that can allow you to actually study in a more localized fashion what's going on. So if you use these enzymes, Fabricator and Fabalactica, and as you can see, then we cut different parts of the antibody structure. It's quite interesting that the Fab arms, basically, of the which are still connected here, of the antibody, they actually really reflect difference in the structural dynamics. But it is not something we can detect if we just look at the at the other half, let's just say, of the FCs of the antibody. So this is just to show that with the right enzyme strategy, you can see how far these events are really decoupled, or how far there is actually crosstalk. So you can localize the conformational effect here. And then to take it one step further, and actually these are now collision-induced unfolding profiles where we challenge an antibody. We basically activate it, heat it with collisions, and we see how stable ion mobility defined conformation is. So here we start off with very much the same collision cross-section, but then we simply qualitatively ask how far are these easy to unfold. So it's a bit like a TM shift analysis. And these are not even the prettiest traces. We've got better ones, but this is done on a Synapse G1, and these are quite large for the low gas pressure we have in that ion mobility cell. But it doesn't matter here, because again, we find at the intact antibody level, wild type, TM, YTE, and TM YTE, we do not really easily see something much different. So the global unfolding dynamics seems to be somewhat the same, but I think we're just not resolving the differences. While quickly going now, because you know where this is heading, I guess, looking at the different parts of the antibody. So for example, the FCs, and you can see in a quite simplistic view that as soon as we have the TM mutations close to the hinge, we really get a much earlier transition to the third, to the most unfolded state. So these two are very similar, and they both share the TM mutation, while the YTE mutation, which is also further away from the hinge, has less effect. Again, it allows us to locate these effects more, and ultimately we can take that further if we can develop the molecular dynamic simulations to match what happens during collision-induced unfolding. And then just to show the counterpart, so if we now look at the sort of most interesting part, the Fab arm, I guess, of an antibody, the business end, then you can again see, if you just look globally at it, that the TM is the more important of the set of mutations, because it actually has the opposite effect here. It stabilizes unfolding. It takes a higher collision voltage before you unfold that part of the antibody. So there is a lot more you can do with that, but I wanted to highlight how, with a wide combination of enzymatic strategies, then middle down, native middle down, I guess fragmentation, but in this case, unfolding studies, you can learn quite a lot about the biophysical behavior of these antibodies. So the summary is that we don't see directly, just from collision cross-sections, what global differences there are, but as soon as we cut it up into parts, we do, and we can localize them. So the hinge, we can see that requires less energy to unfold, but the Fab2 with the hinge still attached, so if you take the other half basically, or cut in a slightly different position, I should say, then it requires more energy to unfold. So it might just be that it has cancelled out for the whole antibody, but unfolding is never in one step, not for such structures. So we can actually now start to resolve the steps. And we can also see what we knew from thermochemistry. We see that also in mass spectrometry, that the stability changes. So we see shifts in thermal stability, or in this case, collisional unfolding stability. So in fact, it's a complementary approach, but it is potentially also a lot faster, and you need less material. So it's actually something, I guess, pharma companies are starting to be interested in. Certainly with AstraZeneca, we are exploiting that a lot further now with different formats of antibodies. Okay, and then I've just structured it a bit according to methodology, since we're in a mass spec group here. I want to talk a bit more about what we can call the labeling methods. HDX is to some extent also labeling, but I will not say too much about it because I think it's quite well known and used for epitope mapping. But I want to dwell a little bit on FPOP and other types of hydroxyl footprinting. So what we can do here is we can also detect indirectly structure and interactions, because we label, digest, and then based on the labels, we can reconstruct structures. But it allows us, and this is much more difficult to do with native MS, it allows us to work in excipients, which means complex buffer systems, which are used when these antibodies are formulated, for example, to actually still study their structure. And also we can then study an effect of the formulation itself. And those are things where it is useful to have native labeling in these complex buffers, but then not worry about it anymore, because we can simply digest and do classic proteomics analysis without having to worry about keeping it on native at that stage. What we tend to get, of course, depends on the method. So in HDX, we get the degree of H bonding, so that's secondary, tertiary structure, but also solvent accessibility, otherwise you don't get exchange. While in FPOP and other covalent labeling methods, we all assume that we are strictly only looking at solvent exposed surface area, no matter what the structure locally is. So they're not quite the same, but they can be quite complementary, these two approaches. And HDX is very popular for epitope mapping. In fact, there is now, just in the last 1 or 2 years, really big interest as well in FPOP and other techniques for that purpose. Very briefly summarize HDX and FPOP side by side. So HDX is the amide backbone. Labels are not permanent, so you have to use specific proteases, digest fast and analyze fast. But I think Agiris will also tell us much more about HDX in a membrane protein context later. The timeline can be as good as milliseconds. Now that's not standard yet, but AstraZeneca, our collaborators, and also J.J. Phillips, we also work with him, University of Exeter, they have together developed a system which is about to be commercialized for millisecond HDX, and it's quite interesting to see what happens at these short timelines. Whereas FPOP labels with oxygen radicals, and it does that in a side chain. So there can be many sides, not just maximum one per amino acid, there can be more. Proteases, you can use any you want, because once you've labeled, you can just put it into any kind of proteomics workflow. So you can also label in principle in cells, but certainly in very complex systems, which is hard with HDX and not done typically. Rather than measuring a mass shift, as you do with the isotopes in HDX, here we quantify based on liquid chromatography profiles. And we then, of course, use MSMS to verify that there is an oxidation and where. And we can even quantify. So we have single amino acid resolution, but we can also quantify, not just say protected or deprotected, but we can say to what percentage. And the time resolution is as fast as microseconds. So that's why in principle, FPOP is actually really quite interesting. In practice, I'll come to that briefly at the end. Of course, it's still not very routine and takes a lot of time at the moment. Examples, I mean, this is just from literature. It's actually from a thesis of one of my students, where someone, in this case, Lisa Jones, had published FPOP data, and for the same antibody, someone else had published HDX data. And it's just to show that when you want to use either method for epitope mapping, you kind of really get the right areas. So you really have a choice. It's more practicalities and what people are used to, what you would use. But they do match the here in this case, because I guess once you do binding, you're really protecting part of a surface. Then you're not looking for allosteric effects either. So HDX and FPOP really give you more or less the same answers. But we used FPOP in a different way, and that's work by Sammy Lawrence, who is just writing up her thesis. And she's been looking at an FPOP workflow to look at dynamic changes in protein structure. Now, I'll show this in a second, but just to explain FPOP briefly. So you take a UV laser, 248 nanometers, and you add peroxide to your sample, and the UV laser then triggers oxygen radicals, hydroxyl radicals, and they react with a protein. But you add a scavenger, normally just a free amino acid, to the solution, and you might need to optimize the concentration because you want your radicals to be short-lived enough so that the reaction is pretty much over in microseconds to milliseconds. And without scavenger, you of course get oxidation, but you don't want to over-oxidize because then the protein could start to unfold or react to the oxidation. But you also, like I said, you want to limit the lifetime of the radicals. So that's something you need to tune, because if you have a buffer, which in itself already scavenges a bit, then you don't need to add so much extra scavenger. But then when that process is finished, so you can, in the simplest case, just pump that through a capillary, but we'll also hear more about FPOP later. But you just end up in a container, can even just be an Eppendorf, and there you have quench conditions, which means a catalase, which actively destroys remaining peroxide. But you also, again, normally add something like very similar to the scavenger chemical, so like an amino acid, which also helps to mop up radicals. And then at that stage, your reaction has stopped and you have labeled your protein. And I should say the labeling is very sparse. So you probably get not more than half of your total protein into a single plus 16 peak. So the majority of the protein is even unlabeled. That is to make sure that we are not denaturing the protein. And that 1 plus 16 peak, of course, means that the position which is oxidized can be pretty much any in the exposed surface of the protein. So you can see that we have a challenge in finding this kind of PTM site, this oxidation site. We need very sensitive and accurate quantification for mapping low-level oxidations, which in principle can be on any amino acid. And we are not just looking for single oxidations, we're even looking for multiple oxidations. And there can be not just plus 16, there can also be other masses, but we have an idea what to look for, but it's quite a list. On the other hand, you can use any enzyme you like. You can do trypsin, you can use combinations, and you do some really good PTM mapping workflow with instrument of choice. But then you also have to put some work into quantifying, because what was 1 peak before, unmodified in chromatography, becomes, just for the plus 16, becomes several peaks, because within that peptide, you most likely have more than one site. And sometimes within one amino acid, you have more than one position. So it can happen that 1 peak before becomes 6 or 8 or 10 peaks after, and you also want to quantify them and verify them with MSMS data. You need to semi-manually check so that you're not misinterpreting. Just because it's plus 16 doesn't mean it's the same peptide oxidized. Just to give you an idea why it's not so routine yet. Okay, and then in a case where Sami used this method, she did actually use protein and wanted to understand how when you have flow conditions, one of the stress factors for proteins is not just heat, it's also frictional forces, when, especially in manufacturing. When you have a whole fermenter full of antibody production and you pump solutions around, and it's known that that can aggregate protein. In fact, in our own blood, it can happen that thrombin triggers aggregation, but that process is tightly controlled. So of course, there is a bit of a switch there, but aggregation of protein in a liquid which flows is an important question. So what she did there is she looked at antibody formats which were developed within AstraZeneca and they're published, but people in the industry call that developability. Because when antibody products are made, and of course many cancer antibody drugs are now really leading pharmaceuticals, one single fermenter batch can cost as much as 120 million US. And you kind of don't know how much aggregation to some extent you will get at the end. Hopefully none, because if there is some, you can't use it, right? So you might have that $120 million question when something has aggregated, or it just happens frequently, or some percentage is aggregated, how you can avoid that. And so you try to engineer hotspots for aggregation out of the antibody without affecting what it does. And so one antibody AstraZeneca had developed called WFL, it's just based on 3 single residues in the whole antibody. It was functional, but it had the issue that it very easily aggregated. So with a lot of work, experimental and also with protein engineering, which means in silico, they made a format of the same antibody called STT, where they basically changed the 3 residues, and you can see that the hydrophobicity has been engineered out. And so these are now great models for us. And in fact, there's a third one, but we care less about it. Even though we still have some hydrophobicity here, they're great examples for basically having switched an antibody from easy to aggregate to hard to aggregate while preserving the function. But we want to understand the biophysical basis of this. So just to prove that this is really working in HPSEC and in a technique which is used a lot, self-interaction nanoparticle spectroscopy, where you look at particle formation. If you just focus on the first 2, you can see WFL is not great, and STT really shows very little tendency to aggregate. But aggregation is a fundamental question. It happens in amyloid as well. And we think there is, of course, some first step, some kind of unfolding step or some activation step before aggregation happens. So can we even understand these early transitions? Can we understand what's going on? I think that's the challenge here. And so for this flow-related question, this works with a system where we can not just simulate flow, but we can physically expose a protein solution in a setup which is ultimately quite basic with 2 syringes. And you can actually then have shear or strain forces around the point where, for example, a diameter is changing, and where there's an accelerating force which ultimately pulls the protein. And you can control that, and you can also calculate these flows, and you can, if you know how to simulate fluid dynamics, you can predict what forces these proteins are exposed to, and then you can correlate that with what you can find. So if you do that, and I'm just showing some highlights because I'm also in the last 3 minutes maybe now of my talk. If you do this extensional flow-induced aggregation study, then just for one single example here, you can see that the WFL, which was aggregation prone, is actually modifying quite differently with and without flow. So it responds much more to flow, while, for example, this STT format does not. So it's much more robust. And if you do that for every peptide or even every amino acid where you see changes, you can start to paint a picture of where the dynamics is affected in this scenario. And then just to say a bit more about the FPOP workflow. So you have single amino acid level quantification, but you need to do that based on chromatography. So again, here you have an unmodified peptide, and you see how many new peaks you're producing. Each one needs to be assigned. They're all plus 16 peaks, but different positions on different amino acids. And then minus and plus flow, even though you have the same peptides, you get significant abundance changes. And you do that all in replicates, of course, as well. So then you have a basis for quantification. And that requires softwares which are not really made for this. We have a lot of softwares now we can use, but it still takes weeks, if not months, to do whole antibody FPOP at the moment, which of course is something we would love to change. And we're working on that. Very briefly, you can also look at effects of excipients such as polysorbate, which is used a lot And you can still label in these, so you can actually start to study the whole system, not just a protein in isolation. Then very briefly, I will talk about some more work from Melissa, where she looks at bispecific antibodies. So you have two basic formats, but instead of having the two pure antibodies, you make a bispecific where you mix these so that these antibodies are targeting two epitopes. But the question is then, what format would you use and what efficiency does it have? So if you change these positions, it has an effect on how well it engages with a target and function is affected, but this is not understood how that translates from the architecture of the antibody to the function. So there we can also, if we focus on the CDR regions between the monoclonal, the pure one, and the bispecific formats, we can see that the accessibility is different. Mel has also done some really cool statistics and looked at what types of residues are predominantly oxidized, because in principle all 20 can oxidize, but some are easier than others to oxidize. And that actually changes as well depending on the local environment. And what I shouldn't forget is that we also need to look at multiple oxidations within a peptide, because what we're doing here now is we are going to make life easier, and we've actually tested this now. If we do not bother to look always at a single amino acid, only if we want to, but if we go back one step and do peptide level oxidation analysis, we can actually get HDX-like data quite quickly. So we circumvent a lot of the issues with the quantification. And we can then do that also for the single, double, and triple, because within a peptide, you likely have more than one site. And you can really see how different formats then react in the CDRs here quite differently. So FPOP, why don't we all do it? Well, it has some advantages, which I mentioned, so I'll save some time by just focusing on the disadvantages. It's still largely manual and multi-step processing, different softwares going from LC to MS to MS, MS, and back. We have radical chemistry, so we are not only expecting +16. We need to be aware there can be other types of oxidations. And of course, if our MS doesn't show us every single residue, then we don't have residue level for every part of the sequence, but we can always dive in and try to pull it out. It's definitely higher spatial and higher temporal resolution than HDX at the moment. There are other ways to do hydroxyl footprinting. FPOP is done by a couple of groups in the US, and then I think so far in Europe, it's only Leeds and Prague. And you will hear from my colleagues in Prague later. You can use cold plasma electric discharge as well, and you can use synchrotrons. And this is actually currently in development at a diamond synchrotron in Oxfordshire, that we will be able to also do hydroxyl footprinting there sometime this year, which will be exciting. Unique advantages there are that you don't need to add peroxide, so you can probably more easily work in cells, tissues, and organisms. Radicals are generated in situ, not added, as it were. You can do dosage experiments. You can actually expose it longer while the laser flashes are constant length. And you can, for example, take viruses in a frozen sealed container and put them in the X-ray beam, and also label the oligonucleotide packing inside the virus. Okay, I'm over time, but I had a bit of a delay to get started. Apologies. I would like to thank particularly my sub-teams, so the first 5 names, 2 have moved on, who work on antibodies, then our mass spec facility, colleagues in the Asbury Center in Leeds, and in particular also the AstraZeneca team, who are very interested in the fundamental science behind what antibodies do, biophysics and structure and function, and all these funders, and you for your attention.
Thank you, Frank, for this great and very informative lecture. There are no questions in the chat, but please, everybody, feel free to type into Q&A chat. And with that, I'm passing the floor to Peter Mann.
Okay, thank you, Michael, for introduction. So good afternoon, everybody. During my today's presentation, I will guide you through some advices how to improve your HDX performance when you're working with antibodies using HDX mass spectrometry. So in the beginning, just briefly summarize the overall workflow so we can highlight the crucial parts. So in HDX, you take a protein, put it in deuterated solvent, and then at predefined time points, you will stop the exchange, and that in that way preserve the label. And to do that, you have to lower the pH, lower the temperature. And then to reach the spatial resolution, you want to proteolyze the protein. And after that, you're analyzing your samples on a peptide level, and you are getting this like iteration over time.
Sorry, Peter, are you screen sharing? I don't see your slides.
Sorry. Is it now on?
No, I don't see. Yes, now,
Yeah,
yes,
now it's
but I
on?
think you have
Yeah.
to go back if
Yeah,
you don't
no
mind.
problem. No problem. Yeah, yeah, yeah. So
Okay.
I will start from that point. So in HDX, we take the protein, put it in a D2O, and at predefined time points, we then want to stop the exchange. And this we do by lowering the pH and lowering the temperature. Then we proteolyze the sample and we run the analysis on a peptide level to get the iterations. And like that, we get the spatial information about the structural changes. And we will now, in this presentation, go through the specificities of these two steps, where we are stopping the exchange and then we are proteolyzing the sample. And we have to balance two things. One is that we have to prevent the loss of the label. As Frank nicely introduced in HDX, the labeling is not stable and you are losing that over time. So you have to keep the pH low, temperature low, and you have to run fast. But at the same time, you want to digest the protein efficiently. And not only to get some peptides, but you always want full coverage. So you have information about the entire protein structure. But you also want to do this with reasonable spatial resolution, which means that you want reasonably sized peptides that are in length of several amino acids. Because you are using non-specific proteases, you are producing overlapping peptides. And we are also looking for some reasonable redundancy. That means how many times each residue is covered, because that again can like increase the spatial resolution, so we can recalculate the iteration from the individual peptides into shorter segments. And to keep a balance and have this like very nice proteolysis for some protein targets can be difficult. And that also applies to antibodies, which are considered as challenging analytical targets. It's not the size. We don't carry that large because in principle for HDX, they are not really that complex. They are composed of like small protein, then reasonably sized protein. The problem is that the whole structure is quite compact and is stabilized by disulfide bonds. On top of that, we have an glycosylation there, at least here for the IgG1, which we are looking at. And therefore, it might be difficult to handle that. And keep in mind, we have to practice structure under HDX conditions. So work fast, low pH, and low temperature. We had quite some experience in HDX and also solved plenty of proteins that were having disulfides, were heavily glycosylated. And yeah, the experiments were working very well. So we were really surprised that when we then came across the antibody, that, okay, for the heavy chain, it was working fine, but on the light chain, we discovered that we are having gaps and we are having some problems to cover the N-terminal part. Surprisingly, when we went through the literature, we found out that this is the case for plenty of other groups, that usually the light chains are quite resistant to the proteolysis in the N-terminal part. And since you want full coverage, that presents like an obstacle in your attempts. So we call that EPIC antibody light chain fail. And we will go through steps that will guide you to slightly better pictures that should look like that, or even pictures looking like that, which is definitely satisfactory. So if you work with a compact and stable protein with disulfide bonds, so you can either leave the cysteines be and hope that it will be digested well, and you will have at least peptides in between the cysteines, which doesn't work always. So then you are having big gaps in the sequence. So we leave this possibility out. And then how to systematically approach such proteins. So the best way is to compare the non-reduced and then fully reduced state. So you perform the reduction before the analysis. So then the structure, of course, is not native. This is not the state which you want to evaluate from the structural point of view, but just analytically. And then this shows you at least which peptides are then generated if the protein is completely reduced. And once you have these two controls, you can go to like HDX-like conditions. And here you definitely want to combine the reduction with the denaturation. And for the reduction, you have to use phosphines because they are working under the HDX conditions. So forget about DTT or mercaptoethanol. Unfortunately, TSEP, one of the phosphines, is not also working well at a low pH. As you can see here from the graph, the efficiency goes dramatically down. So you compensate that by including higher concentrations, which are usually in hundreds of millimoles in the final solution. And for the denaturation, you have several possibilities. So you can either use guanidine, which is highly potent. Unfortunately, sometimes can cause problems in LC because you might not be able to remove it completely, and then you have some adducts in the SI, which definitely lowers the sensitivity. And we know that it also negatively affects the protease. It then tends to create larger peptides. Then you have urea, which is slightly weaker denaturing agent, but the good thing is that it's LC friendly, and we know that also the proteases are quite happy in that. Sometimes they have even higher activity. And if you are from the old proteomics school, then you recall what we were using for the 2D electrophoresis, and that was a mixture of urea and thiourea, which gives you a very strong denaturing conditions that are LC compatible, LC fine, and also it affects the protease to some extent. And using these conditions, you then play with the reduction time. So you let the protein in those solutions with TSEP for 1 to 5 or 10 minutes on ice before you analyze that. And you are trying to approach the optimal picture that was gained through this control with fully reduced sample. There is yet another possibility, and that relies on the electrochemical reduction. That's something that was introduced by Casper Rand. Here, unfortunately, you can't use denaturing agents, but Casper is claiming that they are not in fact necessary. Yeah, so I would love to test that. The only drawback here is that it's quite expensive solution. So once we know that we have to evaluate several different conditions, the question is how to evaluate them and compare them and select the best ones. So in HDX, we are creating the maps of coverage. So if you are comparing two conditions like in here, it's quite easy to say that the conditions represented by the red peptides are definitely better. But if you have multiple conditions and the changes or the differences are not so striking, then it might be quite difficult to pick the right ones. Yeah. We also use sort of parameterization in the field. So we usually refer to sequence coverage. We always want that full. We also refer to average peptide length, the redundancy. That means how many times we are covering each residue. So let's now have a look how that works with the light chain of an antibody. Here we are comparing 4 different conditions. So reduction under HDX conditions without denaturing agents, with guanidine, with urea or urea thiourea. And you can see the numbers for the individual conditions. So definitely here, no denaturing agent means that we are not having full sequence coverage. Yeah. The redundancy, the average redundancy is a bit lower than for the other conditions. Also, the number of peptides is a bit lower. But we are having slightly shorter peptides, which is because the denaturing agents are affecting the protease as well. Might be perhaps more convenient to look not on the average values, but at the distributions. So these plots are showing, in fact, the redundancy. That means how many peptides you have like on each protein segment. And here you can see what we observed on the initial maps, that without denaturing agents on the light chain, you are nearly completely missing coverage in peptides in the N-terminal half, while with the denaturing agent that improves quite a lot. You can also take a look at the graph that is plotting the peptide length, so not only looking at the average number. And that again highlights that, okay, we have a shift. These are the colored ones versus the black one. We have a shift toward the longer peptides, which is not what you really want. But in exchange for like better coverage and better redundancy, we take that. These graphs were, however, drawn based on the LC-MS data, which is like the initial step in HDX workflow. And we have to say that not all the identified peptides are the ones you can then use in the final HDX, because during the deuteration, the intensity goes down. You have wider isotopic peaks, so some of the peptides are going to the noise and won't be useful then for the readout of the deuteration. And to somehow approach that state, we are using what we call a thresholding in the export of the LC-MS data. And here you have the graphs when you set quite a strict threshold, but that somehow reflects how many peptides you will then most likely see in the HDX and providing the data. And you can see that the numbers are going drastically down. And especially for the conditions without the denaturing agents, we are completely losing coverage in the N-terminal half. But also with the denaturing agents, it's not so optimistic. So how we can now improve that further? And what we can do is that we can use the other proteases, other protease columns, because all these data were just using a pepsine, which is like the golden standard and the first choice usually. Luckily, we have now other alternatives. So we have either the proteases from the carnivorous plants, like the nepentazines, or neprosin, or aspergillopepsin, rhizopus pepsin, or Albert Heck's group introduce ANPEP, which is more specific for alanines and prolines. We are typically not using these proteases for the digestion in solution, but we tend to switch everything to the digestion online. So we immobilize the proteases and use a flow reactors, protease columns. The advantage is that this setup is highly tunable. So you can use a column with a single protease. You can couple two different proteases, or you can create a column with a mixed proteases, which is now our like favorite setup. And as I said, tunable, that means that you can also tune the digestion through the flow rate, because that dictates the digestion time, the dwell time in the column. And you can play with the temperature. For the overall analysis in HDX, you have to keep everything at low temperature. But if you go for the digestion at 15 degrees, the loss of the label is very small, but you can dramatically enhance digestion performance. And also the dwell time on the column is in the time of seconds. So it doesn't matter that much that you are increasing the temperature for a few seconds. Let's now have a look how the proteases are improving the overall picture. I won't go through all the possibilities, but I'm just picking the three perhaps most important ones. So we have again pepsine, and now we see how mixture of pepsine and nepentazine 2 or pepsine and aspergillopepsin is largely improving the picture. Also, from the point of view of a peptide length, we now see that the graphs are centered more towards the shorter peptides. And that applies not only to the light chain, but also to the heavy chain. So the only thing which now remains a bit unsolved is this part on the heavy chain, and that actually is the glycosylation side. So the question is how you can handle glycosylation under HDX setup. So again, you can leave that uncovered. We don't like this option. So you can then try to cover that with the glycopeptides. And to do that, you again perform deglycosylated protein. So you can use either PNGSF or EndoS2 in case of antibodies. For other proteins, you can perhaps use EndoH. It depends on the type of the glycan. Like that, you identify the peptides that are generated around the glycosylation site, and then you put that together with the glycan that should be there, and fish those peptides in your data set. And then you can analyze their HDX pair. But luckily for us, Casper and Anesek and Josef Vogelmeier were pursuing acidic PNGases, enzymes that can cleave off the glycans under HDX conditions. And we are quite lucky in that, because we are now together with them developing columns that will allow us to handle these modifications online in the HDX setup. So I think that we saw some quite nice examples that for antibodies, it's really crucial to finely tune the post-quench conditions and digestion step. I think that the light chain of the antibody can be a good indicator whether we handled the whole stuff properly. And it's also good if you want to evaluate number of conditions to have some reasonable workflow that allows you this comparison. And because we don't want to go through all these Excel tables and Excel graphs again, we are now developing the software that allows us, and we hope that soon we will be able to release that, so it will allow that to anyone else to evaluate the digestion, draw the maps, draw the graphs, get all the parameters, work also with all the like possible replications, and that will then move you to about very nice pictures in HDX, where you can very nicely and fully cover the whole structure of an antibody. Surprisingly, it's not applicable only to the antibody, but also to the targets. Here we are looking at the HER2, which is the target of that antibody, of Herceptin. And that protein is also pretty difficult. It's one of the toughest we've ever worked with. And as you can see, the red colors are showing the cysteines in there. And again, you need very proper reduction in denaturation to access the structure and the HDX condition, and then map the epitope of the Herceptin. And with that, I would like to thank all my collaborators and you for your kind attention.
Well, thank you, Peter, for this great overview and also for honoring the time schedule. And with that, we can pass to Maurice Den Boer, who's going to take us back to the intact and top-down. Thank you.
Yes, thank you very much for having me. Let me quickly get my slides. Everything working correctly?
No, we don't see your slides.
You do not see my slides.
Now it's starting. Yeah.
Okay.
Yeah, good.
All right, so good afternoon, everyone. My name is Maurits, and I'm a PhD student in the lab of Professor Albert Heck in Utrecht, where I work on MS-based technologies for characterizing antibodies. So today I want to talk with you about native MS, and then specifically about intact and top-down analyses of antibodies. So what you will learn in the next 15 minutes or so is what is native MS, what can you do with it, and the same for top-down MS. So what is top-down MS, and what kind of information can you get from this for your own kinds of experiments? So let's begin. First of all, an overview of native MS. How does this work? So native MS is a tool that allows you to study intact folded proteins and also protein complexes by MS. So you may all be very familiar with the kind of standard proteomics experiments, whereby a complex protein sample is digested into smaller peptides, which you then analyze by LC-MS in organic solvents. And this can all be done quite robustly. and reproducibly, because peptides are generally not that delicate. So native MS is very different from this. So in native MS, we analyze purified proteins instead, and we put them in a volatile buffering-like solution, something like ammonium acetate, for example. And in this solution, the structure and the interactions of the proteins, they remain intact. Then through gentle ionization of these proteins by electrospray, we bring these proteins into the gas phase, and we are left with an ionized protein or protein complex that we can measure. By then carefully balancing the instrumental parameters, which we have to tune and which we have to optimize for each protein, we make sure that the complexes and the proteins themselves, they stay intact. We send them to the mass analyzer. In our case, that's often an Orbitrap. And this mass analyzer then measures the mass to charge ratio of the ions, here an IgG antibody. And then you get this beautiful mass spectrum, which you see on the bottom right, with the peaks corresponding to the different charge states of this IgG antibody. And from this, we can calculate the mass of the antibody here, about 149 kilodaltons. So that in a nutshell is basically how native MS works. But we can also do some more tricks with the ions, from which we can obtain more information, which we will get into later. So for the purposes of this webinar today, we are going to delve into two specific applications of native MS with theory and also practical examples. So firstly, we're going to look into intact mass measurements, from which we can already extract a wealth of information about antibodies, for example. So we're going to discuss how we can use native MS to help us assess antibody microheterogeneities, such as glycosylation, to study antibody-antigen binding, and also to look into interactions between antibodies and also between antibodies and other proteins of the immune system in forming larger immune activation complexes. So for the second part of this webinar, we're going to look into top-down fragmentation, also of antibodies, by which we dissociate the antibody complex or even fragment the amino acid backbone of the antibody. And this can then give us additional information. And using this information, we can, for instance, validate the sequence of an antibody. And we can also localize PTMs, for example, or other chemical modifications. So first of all, let's start with antibody microheterogeneity. So in the analysis of very small variations in antibodies, there are very few technologies that can rival the level of insight that you can get with native MS. So when I talk about these varieties, what I mean are, for example, things like glycosylations, whereby different glycoforms can be present, oxidations, for example, and maybe also other small PTMs, which all result in a very small mass shift that we can measure. And this is very important for us to monitor this, because these characteristics, they can differ between the production cell line that you use for your antibody and also the specific fermentation conditions, for example. And especially if you want to make a therapeutic antibody out of it, this is very important because these small variations can have a very large effect on the fate and your function of your antibody. So how do we do this by native MS? Well, first of all, we need a very high resolution instrument, which is why we often use Orbitraps for this purpose. If we want to look into the glycosylation of an antibody, for example, the first thing that we usually do is to deglycosylate our antibody enzymatically. And then if you measure this by native MS, you get a native mass spectrum of the antibody shown in the top left spectrum here, which we can deconvolute to extract the backbone mass of this antibody. So that's just the mass of the polypeptide backbone, excluding the glycans, because we have removed those. Now we can compare this backbone mass to the original antibody that still has all these glycans on there. And this is what you see in the mass spectrum in the bottom left. As you can already tell probably, the spectrum is quite a bit more complex. So we see multiple species here that are separated, baseline separated by a mass shift of about 150, 160 Daltons. And the exact shifts between those peaks, they correspond to the additions of different sugar moieties to the glycans that this antibody has. So knowing this antibody backbone mass, and knowing now also the masses of the different glycoforms that we have of this antibody, we can now calculate the difference and assign all these different species to the different glycoforms of this antibody, and also know their relative abundance. Native mass can also be used to study antibinding, for example, which you can use to determine the stoichiometry of the interaction. You can also use this with the correct instrumentation to study the affinity of the interaction. So here we have a very practical example in which we analyze an antibody, an IgG1, that binds to EGFR. So in the top mass spectrum, we see this antibody alone, and for just IgG1 antibodies, this gives us a very clear and highly accurate mass of the antibody and the different glycoforms of this antibody. In the second spectrum, in the middle that we see, we analyzed EGFR separately, and here things are a bit more problematic. So EGFR is a very heavily glycosylated protein, and like EGFR, so are many other interesting antigens. So this used to be a big problem because it's very difficult to measure these kinds of antigens by native MS. And this is because in normal standard native MS, these sorts of extreme microheterogeneities, they prevent us from resolving the charge states. So we see a blob instead of the different charge states, which we can use to determine a mass. So this used to be a problem, but quite recently our lab has developed a solution to this. So we measure these antibodies and these complexes by Orbitrap instruments. And one of the things about an Orbitrap is that the intensity of what you measure, it scales with the charge of the ion. So this means that when we measure single ions, we can make an estimate of their charge. So when we do these kinds of experiments by which we measure single ion intensities, we do a form of charge detection MS, charge detection native MS. And this allows us to mass measure even extremely heterogeneous proteins, such as here EGFR. So finally, we also measure the mixture of this antibody, the IgG1 with EGFR, which you can see in the bottom panels. And this told us eventually that the stoichiometry of this interaction is up to 2 antigens per antibody. But we can also take these kinds of experiments one step further, for example, by analyzing larger immune complexes. And this is what we have done in this example project. So for this project, we were looking into staphylococcal protein A, or SPA, which is known to inhibit immune activation by the complement cascade. So SPA is a bacterial protein that is known to bind to the Fc of antibodies. And we hypothesized that perhaps this binding of SPA could interfere with the interactions of these antibodies and the interactions between antibodies that are relevant for complement activation. So first of all, what we did is to see if we can use native MS to measure the binding of this protein, of SPA. And this is what you see on the right here. And this told us that even though IgGs have 2 identical sites, and this protein SPA has 5 separate binding domains that in theory may each be able to bind an IgG, we see that the interaction is only 1-to-1. So we hypothesized, therefore, that SPA sort of wraps itself around the IgG. So next, we use native MS to study SPA in the formation of complement activation complexes. So the complement system is a proteolytic cascade that is activated when IgG forms clusters, or hexamers specifically, that form on antigenic surfaces. And to these hexameric platforms that form, the complement recognition complex C1q can bind to activate the complement cascade and to eventually promote the clearance of pathogenic material. So for these kinds of experiments, we use the mutant IgG1 that already forms these hexamers in solution, an IgG RGY mutant, it's called. And in the native mass spectra of this mutant, IgG with C1q, which is shown here in green on the right, we see multiple charge states distributions that correspond to the different proteins and the different protein complexes that are formed. So first of all the way to the left, we see the free IgG1. Then at around a mass charge of about 9,000, we see C1q. The IgG hexamer is at about 12,500. And then finally, the whole complex of an IgG hexamer with C1q bound to it at about 15,000. Now, when we added SPA to these samples, we saw something that was very interesting. So over time, we found that the abundance of IgG hexamers and therefore also the whole complex of a hexamer with C1q almost completely diminished. What was interesting though is that we found that SPA did not bind to these hexamers at all. It only bound to the monomeric form of IgG1. So we postulated that SPA, it captures the IgG monomers and in this way prevents it from forming these hexamers that in turn can activate the complement cascade, and that this is the mechanism by which these staphylococcal bacteria can evade immune activation. So also for these very kinds of these kind of mechanistical studies, you can see that native MS can also help you with that. So now I want to continue to the second part of the seminar, which is more about top-down MS. So first of all, some background. So in top-down experiments, we dissociate protein complexes or we fragment polypeptide backbones. And this results in product ions. And these product ions, they tell us something about the composition of their precursor. So for example, what is the protein complex made out of, which individual components go in there. And if we push this further towards fragmentation, we can also determine the sequence of a protein, or at least identify which protein it is. So how do we do this? So first of all, what we do is we mass isolate a precursor from our native mass spectrum, the one that we are interested in. Then we can add energy to this precursor to induce dissociation, or even more energy to induce fragmentation. And this you can do by, for example, collisions with gas molecules, but you can also do this through interactions with, for example, electrons or even photons. So if you add enough energy and you start doing this slowly, firstly, the non-covalent interactions start to break. So for example, interactions between different proteins. If you add more energy and use higher energy activation methods, especially, you can also break the protein backbone, which will then snap into two. And these fragments then tell you something about the sequence. So these kinds of experiments can give you very valuable information for your antibody project. So you can use this, for example, to validate your product, making sure that it is what you think that it is. You can localize any PTMs that are there, or for example, chemical modifications that you may have made. And you can even use this kind of information for de novo sequencing of a completely unknown endogenous antibody. So first I want to show you a very practical example of top-down MS in product validation. So in this project that was done in our lab a few years ago, Andrei Dyachenko from our lab, he characterized an antibody drug conjugate. So this ADC, it had its interchain disulfides reduced, and then the cysteines that were made available in this way, they were conjugated to drug molecules. So for which then a total of 8 can be attached to an antibody. So first of all, by native MS, which you see on the left, he showed that this sample was already a bit heterogeneous and that it contains a mixture of different drug loads. So ranging from 0 all the way up to 8 drug molecules attached. And these conjugate loads, they are of course very important to know if you want to make a drug out of this, and also if you want to optimize your production process. So the next question then would be, for example, where exactly on the antibody molecule are these conjugated drugs? And this is something that we can investigate by top-down MS, for example. So in this example that I show on the right here, Andrei isolated the DAR8 form, so the antibody with 8 drugs, and he subjected this antibody to collisional activation. So this first of all resulted in the ejection of a light chain from this antibody, which we see in the left part of the mass spectrum. And based on the mass that he calculated for this light chain, he could then tell that this light chain had exactly 1 drug molecule attached to it, very likely then on its C-terminal cysteine. And of course, you can go much further with this. So for example, if you want to have very detailed information of where your conjugate drug or modification, something you want to have coupled, is exactly, you can use other ion activation types that provide you more fragments of the antibody, which allows you to pinpoint exactly which residues hold these kinds of conjugations. So what I want to show you next is another application of top-down MS in our lab, which we use for obtaining antibody sequence information. So in this project that I want to show you, we use electron capture dissociation, or ECD, to produce very clean fragmentation spectra of IgG antibodies. So normally in top-down experiments, you may be interested in getting as much sequence coverage as possible. So getting as many fragments, as many different kind of fragments ions as you can possibly get, so that you can match all of these many different kind of fragments that you have to a sequence that you already know beforehand, so that you can see if it matches or not. In our lab, we want to do something different because we are more interested in de novo sequencing of antibodies, so determining the sequence of endogenous antibodies. And for this purpose, it's much more beneficial to limit the possible fragment I types that you form, and to produce fragments of the parts that you are most interested in a very selective and predictable way. So for this project here, we analyzed only the variable Fab2 portions of the antibodies, removing the Fc with a protease. And this is because the Fc portion of the antibody, this is constant. And therefore, if you want to determine an antibody sequence de novo, you do not really need it because it's already known what the Fc should be. So by removing this, we focus on the part that matters most to us. So in this project, we analyzed Fab2 molecules of monoclonal antibodies that were produced in IgG 1, 2, 3, and 4 format. And by subjecting these Fab2s to ECD, we could then induce their fragmentation. What we did here was that by carefully balancing the settings of the instrument, we could make sure that we only got a single type of fragmentation, producing, in this case, almost exclusively C and Z ions, which makes it much more easy for us to interpret the resulting fragment ion spectra. So an added bonus of this approach of analyzing antibodies by native MS was that the disulfides also remain intact. So this means that we can also nicely separate the N and the C-terminal fragments. This is because the N-terminal fragment, which will result in the C ion, it will have quite a low mass, while the C-terminal fragment is much heavier because it's still attached to the rest of the molecule through these disulfides. And these C-terminal fragments, they are the Z ions, and we call those bridged Z ions in this case. And in terms of mass, what was very beneficial for us is that there is a zero overlap between them. So what this resulted in for us is that we managed to produce mass spectra that consistent of almost exclusively C ion ladders of the IgGs, of all the subclasses that we measured. And because these C ion ladders are so clean, it's very easy for us, even by hand, to match these sequential fragments to determine the mass shift between them and see which residue they cover. And this worked for both the heavy and the light chains, and it worked for all of the different IgG subclasses that we tested. So for both of these chains, the heavy and the light chains, we saw that the fragments that we got here cover mostly the part between the intrachain disulfide loops of the antibodies. So the fragmentation starts after the ending cysteine of the first loop, and it spans all the way till the starting cysteine of the next loop. And that's because of two reasons. So firstly, because within these loops, you would have to break not one, but you have to break two separate bonds at the same time to produce a fragment. So that's why this hardly ever occurs. And secondly, as Frank also mentioned in the first webinar this afternoon, electron-based methods preferentially produce fragments in parts of the antibody that are most exposed, which the antigen binding part that we are interested in here mostly is. So this was perfect for us because our coverage is now completely focused on the part of the antibody that holds the CDR3 region, which is the most variable part of the antibody, and also the most important in determining antigen binding. So using this approach, native top-down MS eventually enabled us to generate these very easy to read kind of sequence tags, which we have now already used in several projects in which it helped us to fully de novo sequence endogenous antibodies. So to summarize what I talked about today, we have seen that native MS can uncover a wealth of information about antibodies. We can analyze, for example, microheterogeneities. We can study antigen binding and also the formation of larger immune complexes. And using top-down MS, we can also validate our antibody products. We can localize modifications or conjugations, for example, and we can even provide information that can lead to de novo sequencing of antibodies. Now, if this seminar sparked your interest, there are 2 papers from our group that I highly recommend. So the first one is recent review that we wrote on high-resolution native MS, which should give you a nice overview of everything that the technique has to offer, as well as the fundamentals of how it works. And then secondly, a paper in which we benchmark native MS against competing technologies for analyzing antibodies and also larger immune complexes. So a comparison between native MS, mass photometry, and also SEC-MALS to give you examples of what you can do with these technologies and also for which purpose they are all most suited. So with that, I would like to thank everyone involved and also you for your attention, and I would be happy to take any questions during the Q&A.
Well, thank you, Maurice. Yeah, great lecture. So you are not completely off the hook because all the questions are now targeted at you. Yeah, so you can open it also on your screen, but the first question is, which minimum resolution do you need on the Orbi for these studies? Is it 120 or 240K? So obviously it's a UHMR question.
Let me see if I can go back to the, if I can also open the question. So what kind of resolution you need on the Orbitrap for example, studying microheterogeneities, I think. I would say the minimum resolution for what we usually use for intact antibodies, intact IgG1, for studying glycosylation would be 17,500 or 35,000.
Okay, and then let's look at the last question first on top-down. What analyzer was used with the ECD experiments that you showed on slide 11? And what kind of adaptation was done to achieve ECD? So
what kind of instrument was used for this experiment? So we used a Q-Exactive UHMR Orbitrap instrument for this with an ECD cell from EMS ion, which we put into the instrument. And then, if I remember correctly, we measured these samples at a resolution setting of 140,000.
Okay. And so next question, I think you just answered also Tobias's question. Everything you do on the UHMR, correct? In this work, or is there anything else, any other Orbitrap?
Most of these experiments were done on the UHMR indeed. Although these experiments that you can still see my slides, right? So the experiments that we have done analyzing immune activation, we were very interested in accurately determining the relative abundances of these different species. And for this, an Orbitrap can also do that, but it's maybe a bit less suited than a TOF instrument, for example. So for getting a highly accurate masses, we use an Orbitrap. For getting relative abundances of different complexes, we use a Waters LCT time-of-flight instrument.
Okay. And then there is one more question for you from Eric Baez, which is kind of convoluted in my opinion, with my experience with the CDMS. But do you use CDMS for confirmation of CDMS data? Is the experiment you need to do for binding stoichiometry identification? Will you use a centroid-based approach, Gaussian fitting, for the identification of binding stoichiometry without CDMS?
Let me see if I can understand the question.
Yeah, it's a little convoluted. Maybe if you want to, type the answer later, or we can discuss it.
Yeah, maybe I can answer.
Opens a Pandora box of CDMS.
Yeah, I will answer the question later.
Okay, thank you. So, are there any questions from the audience? Anybody raise his hand? Let me ask one question to start the discussion. We still have a few minutes. To Peter Mann. So all these experiments obviously are mostly done with the therapeutic antibodies, which are IgG1. Are there any experiences with other types of antibodies also, Peter?
Well, so far we worked just the therapeutic ones, which were of the same class. These were antibodies against different targets. So we were using like the Avastin or Erbitux, which are against the VEGF or EGF. So that's a different sequence, but structure highly similar to Herceptin or Herzuma, which was like the primary target.
Okay, thank you. So I think we are out of time. So I propose that there are no other questions in the Q&A session. So I propose that we take that almost 10-minute break, and we reconnect at 4:30 p.m. at 16:30. Thank you.
Yeah. We'll be back at 4:30. We can go through your slides, Arturus, if you're there.
Yeah, I'm here. Yeah.
I'm sorry for my mispronunciation of your name, but
Oh, that's fine. No worries. So let me try to.
yeah, if you can just share your screen.
Yeah, I think it should work. Although I had a couple of times some problems with that, but let's see if that's okay. Can you? Oh, yeah, I don't want to see my email, but
yeah.
Can you see the presentation now?
Yeah, I see it. If you, yeah, yes, super.
Okay, that should work. Yeah, okay.
Yeah,
I think that's it. Okay, good.
Yeah, great.
I don't think it will be any problems, so that's fine. Good.
No, I think it should be okay.
All right, good.
Okay, then I will see you in a few minutes.
All right, fantastic.
Okay, bye. Bye-bye.
Yeah, bye.
Martina, just a quick question. Any concluding remarks you want to make, or Albert or anybody else? Sorry, we don't hear you. Is it on our side? I think we can't hear you.
Can you hear me now?
Yes, yes.
Yeah, sorry. I think if you just thank everybody for their support during the 4 years with Epic Access, and yeah, that's
okay. Thank you.
Yeah. I think we can start now. Julian is back, so it's already 4:30. If Michael is there too.
Yes. So welcome to the second part of this afternoon seminar, the second section that's going to be focused on transmembrane protein. And first is Julian Langer of the Max Planck Institute. So we are passing floor to him. I'm stop sharing now.
There we go.
Yes,
be able to see my
screen. Thank you.
Awesome, perfect. So yeah, thanks for the introduction. And for the next half an hour, I'll be talking a bit about structural proteomics of membrane proteins. And maybe as a short introduction, what we do in my lab. So I'm located at 2 Max Planck Institutes in Frankfurt, Germany, and I'm specialized in developing mass spectrometry-based techniques for researchers at the MPI for Biophysics and Brain Research. Now, in biophysics, the research focuses very structural biology-centric, so also that's what I'm going to be talking about today. But we're actually also looking at quite a few research questions in neurobiology, so there we're more omics-focused. So consequently, the methods we develop and we implement actually range quite a wide spectrum. And so on one end of the spectrum, we have these targeted analysis where we work closely with structural biologists who have membrane protein complexes with unknown subunits, which are almost completely inaccessible to conventional bottom-up proteomics, simply because they don't digest well or they don't fly well. They are simply too hydrophobic, and there we develop instruments and approaches to actually still be able to identify these subunits. I won't be really going into details on that today, but if this is something that you're interested in, please feel free to answer me later, or to ask me later. Then, and that's going to be the topic I'll be focusing on mainly today, we're doing functional studies on these membrane protein complexes that we, in general, have structures on. And we use HDXMS a lot, and also a bit of cross-linking, also other functional assays, competition-based assays, and other secondary reporter assays. Today, I'll be focusing mostly on HDXMS. Then we're also doing bottom-up proteomics, the whole show, from shotgun turnover, phosphorylation modifications, and also artificial amino acids that we encode in transgenic animals to look at specific cell types, for instance. And last but not least, a bit of MS imaging. The instruments we use for this are actually quite diverse. So for our targeted analysis, we're mainly using MALDI on a Bruker Rapiflex that we modified. Our HDXMS is running almost exclusively on a water synapse with an HDX2 automation system. And for our bottom-up proteomics, we have a few Orbitraps, Eclipse, Fusion Lumos, Qexactive Plus, and a Bruker TIM stuff. So that just to give you like a bit of background on what kind of stuff we do. So today, I'll be focusing mainly on HDXMS, and I'll be showcasing you 2 example projects that we've recently worked on. The first one is going to be on a proton channel opening in a transhydrogenase, so a prokaryotic enzyme. And the second one is also a prokaryotic enzyme from Geobacillus, BD oxidase, so a terminal oxidase, a part of the respiratory chain that is essential for these bacteria. And there we look at substrate and inhibitor binding. So first project that I want to talk about today, the E. coli transhydrogenase. This is an essential enzyme for many organisms, for many prokaryotes, including E. coli. It maintains homeostasis, or it's one of the elements maintaining homeostasis for NAD plus and NADH and NADP plus and NADPH. So it basically governs the pool of reduced and oxidized forms of the two molecules, and it does so by binding both and by harvesting the proton gradient over the membrane to reduce NADP plus to NADPH and using NADH. So structures of this protein are known, mainly from also eukaryotic homologs. For instance, here's an ovine structure that we use to to plot a homology model of the E. coli transhydrogenase. And Robert Gannon's lab, they actually specialized in this. So they have a few activity assays, and they had pretty distinct conditions where they knew that a particular activity state of the enzyme is active. And we have snapshots of the structure, but we have a couple of points in the reaction cycle that we don't really know how they work. So the main point being how that proton channel opens and which transmembrane helices here in domain 2 are actually involved in that. So you can see that we have different binding states in this proposed reaction cycle that we can track. And for us, we already knew more or less where NAD+ and NADH and NADPH bind, but it was really unclear how and when the proton channel opens and also to what extent. So that's what we wanted to track with HDXMS. And the work that I'll be showing you now on the next slides is mainly by Martin Eisinger and Jonathan Zeller. Jonathan is still a PhD student in my lab. Martin left us 1.5 years ago for AbbVie. So also this is a photo that Jonathan used on his ASMS talk last year, so I have no qualms using it here as well. Anyway, so this is a quick overview of the experimental setup. We have reasonably pure protein here. You can see that the protein has actually no other bands on a gel, but we naturally also did bottom-up proteomics, so it's a really pure protein preparation. We know that in the biochemical environment that we did our HDXMS measurements, the protein is active. So we did that using a secondary reporter assay. We did this in 0.05% DDM. This is a recurring trend that you'll see. So we're trying to use an environment that maintains the functional integrity of the protein, but also gives us a good handle on the protein LCMS wise. And then we use an HDX2 automation setup coupled to a Synapse. So not the most new system, but a very robust system that works very well in our hands. So how does the data look like overall? One of the key elements of a good HDX analysis is sequence coverage. So that determines or that limits how much you can see in the protein. And here in dark green, you can see the respective subunits that we got for the different states of the protein that we monitored here. And you can see that in the transmembrane segments, we have a couple of elements which we would actually like to see. So we'll revisit that later, but overall sequence coverage of almost completely north of 80% is a pretty good starting point to get an idea of what's going on in such a protein complex. So the first data set I want to share now, or the first part of the data set I want to share now, is the NADPH bound state, where we believe that NADPH should lead to a binding in domain 3 here, and the proton channel should be open. And we compare our deuterium uptake that you can see here, that we plotted that to a non-ligand bound state. So it's differential deuterium uptake mapped here. A blue color indicates an increase in deuterium uptake. A red color indicates a decrease in deuterium uptake. And you can see right away that there is the segment here in domain 3, which represents NADPH binding exactly in the location we expected. Loop D, we were a bit surprised that that wasn't more involved here, but this could be, it's actually quite flexible that it is actually involved in binding, but it's just not, our deuterium uptake is just not reduced so much. But this is exactly the site that we know NADPH binds. So that's a good sanity check. And the peptides we actually saw, they were really beautiful and reference uptake plots that we're very, very happy with. And then we are also really excited to see that in several of the transmembrane helices, you can see down here, we observed an increase in deuterium uptake up in NADPH binding. So can actually track now which of these transmembrane helices are involved in opening up a proton channel to get the protons up for NADPH binding. And this is now an example uptake plot for one such peptide from here. So you can see that in the presence of ligand, the uptake is significantly higher than in the absence of ligand. This is the channel that gets opened up. Naturally, and also this is something that you always have to keep in mind when you plot these data, and maybe Argyris is going to go a bit into that later. Here we are plotting on a static structure with a known with a closed channel. So we can only say that these helices move, but the structure we are plotting here is not actually an open form. This is a closed form, but we know that these helices are actually moving apart to let protons through. So anyway, that being said, let's take a look at the other state. So this is again the NADPH state. Then next looked at NAD plus state, and you can see that the NADH binding site is actually up here. So we see moderate binding here, but we see no effects in the rest of the protein, particularly no changes in the transmembrane segments. So that fit the model very well. Then upon NADP binding, we actually had a bit of a surprise. So we didn't, we expected a partial opening of the channel here due to an induced hit here. That's kind of postulated. We're a bit surprised by the increased deuterium uptake that you saw up here, but this could really be due to a lower affinity for NADP+, which leads to a change in the conformation and back and forth motion of these segments of the protein. And we also see that even NADP+ binding leads to or still induces partial opening, but just on one side, on the cytoplasmic side of the protein. And then the last experiment we wanted to do is we wanted to see if now we have a protein in the NADP+ bound form, and we gave it also NADH, so the reduced form of NAD+ that can be used to reduce NADP+ to NADPH. If we could induce this full channel opening, and we're actually really excited to see that that indeed worked, and that we get an increase in deuterium uptake across the other transmembrane helices as well. So that was really beautiful data, in my opinion, at least. And however, when we looked at the transmembrane helices here, there were a few elements where we were still a bit like we would like to see them. So we didn't really have a full coverage or complete coverage here. And, when we recorded these data about a bit more than 2 years ago, we thought about how we could improve them, and we were in close contact with Waters. So they were just about to launch their cyclic IMS system. So we were very fortunate that we got some measurement time on one. And just to reiterate on the improvements of the system, so the main improvement is a cyclic ion mobility cell that gives you more separation in the ion mobility dimension, but certainly an enhanced ion transmission in the step wave is not going to hurt your performance for mapping membrane protein or transmembrane segment derived peptides, which are typically lower in intensity, and also the additional resolution and the more sensitive detector wouldn't hurt. So we recorded a data set from an identical aliquot that we also measured on our Synapt, identical HDX2 automation. And the data overview at first glance was, let's say, a bit underwhelming, not going to lie. So if you look at the peptide numbers we get for the two subunits, those are almost identical, with the B subunit, which is actually more membrane embedded, a bit higher, and but the sequence coverages were rather identical. Again, here is we have a bit of an improvement in the B segment. So that was a bit, yeah, we said, okay, but let's take a closer look at the data. And we actually saw that the data are actually quite exciting and quite good, and that we can really gain something. So if we take a look at a couple of example peptides, so here in the soluble segment up here, near the NAD+ or NADH binding domain, you see that we actually have much cleaner spectra, but the intensities we don't really gain all that much for this peptide. However, if we then look at a couple of transmembrane peptides, such as this one, on the left-hand side here, you see the Synapt data. On the right-hand side here, you see the cyclic data. You see that due to the also higher resolution ion mobility data, we can clean up the spectrum much better for assigning the isotopic peaks for monitoring deuterium uptake. So that was really that was really exciting for us, and we dived a bit deeper into the data and did a full evaluation. And this is a comparison of the Synapt data and the cyclic data. Also, keep in mind here, please, that we had to adjust a bit the color scheme here. So this was previously a 20% plus minus differential uptake map. It's now minus 40%, because with the cyclic data, we did get higher differential uptake rates. So we are actually more sensitive in our detection, and we were able to track better uptake. So if we take a look at a couple of peptides here, for instance, the one up here, which is not affected by NADPH binding. So yeah, important point I should have probably made here. This is the NADPH plus minus conditions. We wanted to see channel opening if we can see more of these transmembrane helices. So unaffected segment, amino acids 50 to 56, no change, and similar performance, a bit higher overall uptake in the cyclic data, but yeah, we see the same. Now in the membrane protein or in the membrane domain or transmembrane domain derived segments, we see actually that our uptake values are significantly higher for these two example peptides. And we actually had a few peptides in particular back here in team 14, which are unique, which we are only able to detect in the cyclic on the cyclic instrument. These were actually also low intensity peptides, so that really helped us to understand the proton channel a bit better. So we also recently published that end of last year, and that was, I think, a really nice study showcasing the power of how HDXMS can complement a structural biology project by being able to investigate and analyze transition states that you can't really differentiate in an EM or with X-ray crystallography. So as an outlook, next obvious steps are multipass measurements to increase resolution a bit further, in particular going for larger and more challenging membrane protein complex, because we're a bit more sensitive, and that's certainly on our to-do list. So that sums up the first part of my talk, and now we'll change gears a bit, and we'll move on to a BD oxidase from Geobacillus. So this is a respiratory chain complex enzyme, last part of the respiratory chain, so it reduces oxygen to water. We actually solved the structure, or Shahra Zafarian, a member of Hartmut Michel's department in our institute, solved the structure. We assisted that project by identifying new subunits in this that were previously not annotated. It's a potential drug target, and even though we tried quite a bit, we had no structures of the actual protein together with the potential, or with its substrates and with inhibitors. So this was really was a really good and tempting target to do HDXMS on again. So we looked at substrates with, which in this case is coenzyme Q1, an electron donor, donating electrons for oxygen reduction, or orokin D1, which is a fungal derived suicide inhibitor for the BD oxidase. Again, the work now is done by Jonathan and Martin, and also Christina, a former master student in my lab, who then joined for her PhD, but then went over to do more brain research related work. Anyway, so the following data is basically generated by these three. So again, our setup, our HDX2 automation setup, experiments were conducted in DDM again. This is a quick overview of the actual sequence coverage that we obtained. On total, about 170 peptides that we mapped. You see also that our uptake overall, or fractional uptake, was not great. We have a couple of elements that took up pretty well. So those are the soluble segments, but we also have a couple of segments that are relatively slowly, or taking up deuterium relatively slowly, and we also have quite a few gaps in our sequence. This is the unfortunate truth of membrane protein HDX. So you can get reasonably good sequence coverage of 80–90%, but you'll always have a few gaps that are really challenging to get. Anyway, with that being said, here is the data that we got on using our substrate. This again is a structure plus minus our substrate differential uptake plotted in a color gradient from red to blue. So red means lower uptake. So if you want to look for the binding site, focus on the red segment in the structure. And it's actually pretty apparent that there is a big red beta barrel up here, or sorry, not beta barrel, but beta sheet segment up here, which is actually in the so-called Q loop, which was the proposed binding site for the keynoles for the substrates. And it's also directly above the heme prosthetic group here, that is a supposed primary electron acceptor. So that was really we were really excited to see that. That was also an internal sanity check we had in our data. And if we then took a bit of a closer look, this is really that made a lot of sense, seeing a decreased deuterium uptake here, because it's essentially the Q loop clamping down on the substrate going in here for heme B558. And we were though quite excited to also see that there was an increased deuterium uptake in this small alpha helical segment here, which coordinates heme B558, which could indicate a conformational change, pulling the heme up and closer into permissive distance for electron transfer from the substrate. But that wasn't the only thing we saw. You also see these differential uptake colors here in this essential segment, and really excited to see that this actually went along the putative electron transfer pathway, and that it was located down here exactly at the segment where a potential proton channel goes in, or a proton pathway or entry point. It comes in for actually providing the protons together with the electrons from the kenol for the oxygen to be reduced here. So that was really exciting for us, and it gave us insights into the conformational dynamics of the complex up and substrate binding that we couldn't get from the static structure. We then incubated the protein with the inhibitor, so same identical conditions, and this is just a static picture now of the inhibitor binding. You can see the Q loop again up here, and you can see that right now the differential uptake is actually even more widespread throughout the Q loop, and actually that these two beta sheets here are not only the two beta sheets are affected, but also these not so tightly folded segments back here. So it's a suicide inhibitor with a higher affinity than the quinol. So that kind of made sense. And we're really excited also to see that the entire set of differential uptake segments, or differentially uptaking segments we saw here in the central part of the protein, they did not display any differential uptake in the inhibitor form as compared to the substrate bound form. So that really encouraged us in believing that these are physiologically relevant functional changes in the protein complex. So yeah, so this is a data set we've actually been sitting on for almost 2 years now, or a bit more, and we are about to submit this manuscript, but I thought I'd share it with you to really show you what kind of data you can get out of a membrane protein with HDX in this. Now, I'm not sure how much time I have left, but I have 2 bonus slides on a bit of a completely different topic, and this is a bit of actually lipidomics and structural proteomics. And this is a project that we recently went in. I think I have about 5 more minutes. That should be easily okay. So there we studied P116. Name probably doesn't ring a bell. Also didn't for me. It's a mycoplasma protein that was totally uncharacterized previously, unknown folds, no idea what it is. It was purified with the lab of Ignacio Fita in Barcelona at IMBNB, and Achilles Frangakis, cryo-EM specialist at Goethe University in Frankfurt, he actually solved the structure. Actually, one of his PhD students, Lasse Spanke, solved the structure. So this is how the protein looks like. It's basically 2 hand-like protein, or segments of the protein that are a bit folded against each other, and they both have this hydrophobic cavity here. And this was really exciting for us because Lasse saw several densities in this hydrophobic cavity that they could really not make sense of. And mycoplasma are a parasitic organism, and it's not really clear to date how they take up nutrients from the host and how they, for instance, also take up essential lipids. So when we saw this, we were like, this could actually be lipids on the inside. So we first did a really simple, low-tech lipidomics assay just to see what's going on. And we did some MALDI on organic solvent extracted protein samples, either from pure P116, which was produced in E. coli. So it had a lot of E. coli lipids bound to it. But we then emptied P116 with a mixture of detergent and different detergents and dialysis treatment, where we could remove 95 to 98% of the lipid content. And we then incubated P116 that had been emptied with serum, so with serum lipids. And we actually saw that we got quite a different uptake pattern here. So we then decided to go in on this and re-rigged our timsTOF Pro 2 with our nanoelute to do a lipidomic screen. And we actually identified in total more than 500 lipids in this. And now all of these peaks here that you can't really assign unambiguously in a normal MALDI spectrum. In a MALDI TOF, you will not be able to separate them. If you use LCMS, that's a fairly straightforward thing. And if you have iMobility, it becomes even easier. So we saw this as mainly PEs and PGs from E. coli. So stuff that you expect, but that probably has zero physiological relevance because it's just an artifact from protein production. So we then looked at, can we get rid of these protein production artifacts using our emptying protocol, so our detergent incubation? And the answer is yes. So most of the lipids are gone. And yeah, I should have mentioned here the color code for these lipids. So this is all of the lipid classes we tracked. The color code here is from relative abundance of the tick in the sample from maximum to minimum. You can see that we have some leftover PEs here, but that the majority of the lipids have been efficiently depleted. So when we then incubated it with serum lipids, we actually saw that the lipid pattern has changed completely. So we saw a lot of PCs, so phosphatidylcholines, sphingomyelins, and also cholesterol esters and cholesterols, which was really exciting for us. However, when we ran this experiment, we were immediately like, we thought, oh yeah, what if this is just the who's who of whatever is present in serum? So what if this is just the lipids that are present there in a vast quantity, and P116 just grabs whatever is most abundant? So we also did lipidomics on the input sample, on the serum, and we were actually quite excited to see that the lipid distribution in the serum is completely different than what P116 extracted from this from the serum. So this is actually part of a manuscript that's going to come out next week in NSMB. So, but I decided to show it to you now because I'm also quite excited about it. And we're actually going to do structural proteomics on this as well, doing HDXMS to track how these lipids are actually getting extracted from the donor membranes together with electron microscopy. So yeah, that brings me to the last slide, to the acknowledgements. So I'd like to thank the directors at both of my institutes for allowing me to set up shop here. This is the mass spec team that you can see here. We grew quite a bit. So I used to have 5 people. We're now at 9. So it's really great. We're doing quite a few different things. These people are the collaborators that were essential for getting the projects running that I talked about today. Bob Gannis for the transhydrogenase, Charles Zafarian for the BD oxidase, and Achilles for P116. These organizations helped me fund my lab, and these companies were very grateful in their method collaboration. So with that, I'd like to thank you for your attention, and I'm more than happy to take questions later in the Q&A session, I think. So yeah.
Well, thank you for this exciting talk, yeah, and for finishing on time. There is a question for you for later, but now we're gonna pass to Peter Novak.
Good afternoon. Now it's my turn to follow in membrane protein analysis. And instead of talking about transient labeling, I am planning to talk about the stable covalent labeling. That might be also important for analyzing the transmembrane protein. I don't know if you are familiar with the protein footprinting, but originally the footprinting techniques were designed for studying the interaction of RNA or DNA with protein. And I will do only very short introduction in this topic, since Frank gave you a very nice description about this area. But it's probably a good idea to refresh a little bit your memory. I don't know if you got a chance to get a coffee during the break. It was quite short, but I made it. And there are several possibilities how to study the protein conformation using the labeling techniques. One is limited proteolysis, but it's based on the enzyme accessibility of protease towards the protein. But also there are the covalent labeling, which might cover like hydrogen nuclear exchange, but it's mainly capturing the rearrangement of polypeptide backbone. But also there are other 2 techniques called chemical or radical probes that are targeting amino acid side chains. And in principle, what you do, you are dissolving the protein in the buffer that it's nearly physiological, and then you are introducing some sort of chemical or radical probes to decorate the amino acid residues. What's also the difference between the HDX and the chemical or radical labeling? It's the fact that in HDX experiment, you mainly do the nitrogen pulse in different period of times. So you can also monitor the dynamics of your protein systems, but in the radical or chemical labeling, we do just a single pulse at the defined time. So we are mainly getting some things what's very similar to like the regular snapshot in compared to HDX. It's similar like recording the situation, having the camera.
if you need to know more, it's a good idea actually to work with Michael Grossi. What's also important to keep in mind the difference between the targeted labeling and the radicals. If you do targeting, you are mainly targeting the selected amino acid side chains, but in the radicals, in principle, they can target almost all possible amino acids, excluding the alanine and the proline. What's the goal of the experiment? It's to analyze identifying the amino acid side chains that are exposed to the solvents. What you do, for example, if you are interested in the composition of this tetrameric complex, you are introducing very short pulse of the chemical radical probes, then you have time to digest your sample in the pieces. In this case, we are having some sort of advantage because we don't need to worry about losing the probe like in HDX, that we need to cool down the samples and cut it down as soon as possible to avoid the back exchange. You can use the trypsin, lysine, whatever you like at basic pH. And then you do the regular data-dependent analysis, identifying all regular peptides, then the modified peptides. And based on the intensity between the regular and modified peptides, you can somehow design the structure of your protein systems. In principle, similar to HDX, it's comparative approach. So it means you need to analyze your sample in when, let's say, the protein is alone, and then the protein forms the ligand with other protein, or it forms a complex with low molecular ligand. So what you do, you are introducing the labeling piles. The majority are using the OH radicals. Then you are digesting the sample into the pieces by trypsin. Then, of course, you are separating your tryptic peptides on reverse phase. Then you are identifying the peptides carrying the modification. Of course, you need to also identify the regular peptides. And once you have the information, you need to back to your MS trace. And in the MS trace, you are identifying the ratio between the modified peptides and the regular one. And of course, if you are incubating your proteins with two different ligands, you can see if the ligands it's somehow making this part of the protein more accessible to the solvent, which is this case, or is less accessible to the solvent, is the other case. So if you are, for example, investigating these homodimers, and you are putting that the low molecular ligand, which might actually stabilize the complex or favor the dissociative complex, you can easily see it, and then you can plot it in some sort of curve, which may be used for the further molecular dynamics, and you can get some structure information out of it. Of course, there are different ways how to generate the OH radicals. One possibility is the old-fashioned one, is the regular phantom chemistry, but it's quite tedious and it's continuous. It's hard to control it. The second possibility, which was introduced by Marksins, and I remember that Frank mentioned that, it's using the cyclotrons that are dissociating the water molecules. But what's actually the most common way how to produce OH radicals is using the excimer laser dissociating the hydrogen peroxides. Of course, you might introduce some artifacts because the hydrogen peroxide, even without the photons, can oxidize the proteins. Of course, it's not that easy that it looks like, because not all amino acid side chains are sensitive towards the OH the same way. There is some that's very important to keep in mind that cysteine, methane, and tryptophan are more susceptible to oxidation than the tyrosine, phenylalanine, histidine. And as I mentioned before, the alanine and glycine are almost resistant to this kind of modification. The setup is quite simple. You have a syringe with your protein samples in the biologically friendly buffer. Then you have a T-piece, and you are mixing your protein sample with the peroxide. So the protein is not sitting with the peroxide for a long period of time. Then you are introducing just a very short pulse of the laser. And then, of course, you need to quench the reaction immediately by adding the methionine, or you can also keep the catalyzer to get rid of the residual hydrogen peroxide, which wasn't destroyed by the laser pulse. And in the reality, the whole setup might look like this, that you have these 3 syringe pumps and the holder having these few silica capillary which burn polyamide coating, that it's and it's making transmissible for the UV light at the wavelength 248 nanometers. It's very important to know that after the labeling pulse, you have a chance to reduce and alter your protein sample. If it's heavy glycosylated, you can use the PNGs to deglycosylates, then it's dehydrated proteins, and then you do regular LC MSMS data dependent acquisition. And it's very important to know that it's quite complicated to analyze the data. That's something I was planning to show you today, that even you are dealing with very simple protein mixture, you may end up with hundreds of peptides coming just from the single proteins, because almost each residue can be modified. And if you see from this picture, some residues might be modified by a different kind of chemistry, which will actually increase the complexity of the mixture. For example, having the ion mobility separation, it can give you advantage, because, for example, if you have this like the octapeptide, where 5 out of 8 residues are oxidized, and in principle you are forming the isoparic peptides, because each oxidized product appears at the mass H2 plus at 201.78 Daltons. And of course, you can somehow differentiate it by the chromatography. It's also good to have information about the cross-section, because the oxidation on each residue, it's changing the shape of the peptides, which is additional information how to deal with it. And in principle, you can use also the ion mobility trace to quantify the ratio between the regular and oxidized peptides for each residues. And it's exactly what Frank was telling us before, that you can reach the single amino acid resolution during the FPOP experiments. But of course, there are some problems with the FPOP for membrane proteins, because you can imagine that the water doesn't go well with lipid bilayer. Also, the OH radicals are even more polar than water, so it's very difficult for OH radicals to get into the membrane and label the amino acid side chains that are surrounded by detergents or by lipids. And Michael Gross got very clever ideas, and I think he got almost like more than 3 ideas, but I will show you just the 3 of them. First, he decided to use the perfor isopropyl iodine that is dissociated by the laser to form the perfor isopropyl radicals, but these radicals doesn't modify the protein at all. Also, the iodine radical is modifying the aromatic residues. In this case, you can see the single and double modification of the tyrosines. The second option is use the carbon chemistry, and so you can irradiate with the same laser the 3,4 methoxy benzyl bromine. You form these, you find this benzyl radical that can also get into the membrane, because it's a little bit lipophilic. And the last development is based on the titanium dioxide that is mixed with the mid cell containing the proteins. Then it's again illuminated with the laser pulse. The titanium oxide particles are producing the radicals from water or from surrounding lipids, which may be used to decorate the proteins part which is sitting in the membrane. And I will talk about all these possibilities in details now. The reason why Michael Gross picked up this kind of perfor isopropyl iodine is very simple, because he was able to calculate that it might go well with the lipid bilayer. And he found out the tricks that sonication of the sample might help the incorporation of these of the iodine radicals into the membrane. So it's very simple. He was firing the laser on the protein sitting in the lipid bilayer, and also introducing the sonic pulses into the samples. And he was comparing first the data once when the sonication was on or it was off. Once the sonication was off, he was only able to label the soluble part of the transmembrane protein and few amino acids sitting right on the edge of the lipid bilayer. But after introducing the sonication, it was also possible to decorate the amino acid sitting inside the membrane. As a proof of principle, he used the vitamin K exposed ribsidase and the same proteins, which was very convenient, because it was possible to see the differences once the sonication it's imposed to the protein systems. He was able to label several amino acids within the cell membrane, but only once the sonication was introduced, he was also able to modify the residue that are really buried inside the lipid bilayer. The second possibility is to employ the carbon radicals or carbon cations. For the reason he selected these three methoxy benzyls radicals, and the reason is very simple, because they can react very well with proteins. That's visible on these pictures on the G, and also they are very heavy, low. They are carry very low stability in the aqueous buffer, so they immediately stick to the proteins, which allows you again to structurally characterize the lipid surrounding regions of these vitamin K epoxidase reductase. What's the most promising step is using the titanium oxide that is in principle in combination with lysyl pulse able to produce the radicals that might go into the membrane and can modify the part of the protein surrounded by lipids or detergents if you are playing with artificial micelles. But he found out that if he is introducing the acetones, he might actually employ the principle of Paterno-Bush reactions to modify the intra-membrane part even better. He compared the oxidation products of these vitamin K epoxidase in the presence and in the absence of the acetones, and you can. It's very easy to see that in the presence of acetones, the labeling efficiency in the transmembrane regions was much better. At the end, he was trying to compare the conformation state of the GLUT1 transporter, and he was comparing the accessibility of some amino acid sachets in the presence of the glucose. The glucose is a regular molecule that is transported by this transporter, but of course he did this experiment in the presence of maltose, that it's keeping the open position out parts, and also the cischoline that is keeping the conformation open inward. And using this kind of chemistry, he was able to distinguish between these three different states, localizing the position of the one or the other inhibitors. Of course, it's very difficult to play with the excimer laser. There are some safety issues, and it's not so easy to set up every time the same density of photons. And so we got an idea that it might be good to replace this photochemistry with the radical chemistry, with the radical chemistry, and make it simple. We find out there are some nice chemicals designed by Tony at ETH Zurich that once they are mixed with any kind of Lewis acids, so they are introducing the protons. The protons cause the decomposition of Tony regions, and then the radicals can modify any aromatic amino acid within the protein sequence. It's possible to modify the tryptophan, tyrosine, phenylalanine, histidines, and to form the stable covalent bond between the 3,4 alkyl radicals and the amino acid side chains. Of course, it's also possible to modify the cysteine residue, but only in the case when it's in its reduced state. For this experiment, we find out we were playing with different probes, releasing the 3,4 alkyl radicals or releasing the tetra fluoroethyl imidazole radicals. That's a little bit bigger. And we start tuning the conditions to do it also in the quench flow setup and control the time of the reactions. It's very simple. It's possible to mix the protein with the Tony regions in one syringe, and the Tony region doesn't do anything to the protein if the inductor is not there. In this case, it's a vitamin C as a proton donor. Then it's mixed in this TPs with the protein and Tony regions. It induces the 4-alkylation of the protein, which is stopped by the addition of the tryptophan, and it's immediately transferred into the Eppendorf tube, where there is a solution with DTT that is also scavenging the iron molecule, so we are not getting the oxidation of the protein. And the setup is very simple. It's almost the same setup like of the FPOP, just the laser it's missing. We tested these hypotheses on the APO, analyzing the APO and hollow form of the myoglobin. And of course, we were able to modify several aromatic amino acids within the myoglobin sequence, and comparing the labeling efficiency in the presence and absence of heme, we were able to identify these amino acid chains that were heavily modified in the absence of heme. And it's not surprising us, once the heme is released, then the flow allelers got a chance to get inside into the binding pocket for the heme. And of course, we were able to identify some residues that are more accessible to our radicals once the heme is there, because the heme binding, it's making some allosteric effect to the rest of the myoglobin molecule. We compared our data to the FPOP data, because it's also some sort of the radical chemistry, and we got a nice overlap between the FPOP chemistry and our 4-alkyl radicals. In the next scenario, we decided that it might be good to try this radical chemistry for transmembrane proteins, because we believe that the any like 4-alkyls are very lipophilic particles. So we started with cis-prenyltransferase, and it was a nice collaboration. It's a nice collaboration with our friends from Israel, from Moshe Giladi and Yoni Haitin. They supply us with this cis-transferase. It consists from 2 different proteins. It's a NOGOB receptor, and it's also, and that's the one part. And the second part, it's the dehydro diphosphate synthase. And it's very important in the glycosylation of the proteins, because it's responsible for synthesis of dolichophosphate that is further used for glycosylation of the proteins in the plasmatic reticulum. We apply the same kind of chemistry. We started again with the small probes. It was again induced by the vitamin C. Once the 3,4 methyl radicals was released, that it started modify aromatic amino acids. We're able to detect several amino acid residues modified by the 3,4-methyl. And of course, we did again 2 experiments, since I was talking that you need to do like differential experiments. The blue bar represents the protein alone, and the red one represents the proteins with the farnesyl pyrophosphate or farnesyl diphosphate. It doesn't matter, which is a natural ligand for this. And of course, we're able to detect 4 residues that are less modified once the farnesyl pyrophosphate was present. It was possible to plot all these modification on the structure of cis-prenyltransferase. The residues label are modified by our 3,4-methyl radicals. And of course, once the farnesyl was added into the reaction prior the modification step, it was possible to see the degrees in the 4-alkylation of the aromatic amino acids, such as they are in principle in the near proximity of the binding pocket for farnesyl, which means that this kind of chemistry allows the detection of the binding pocket for the farnesyl or geranyl. The second step of our effort is to test to increase the spatial resolution of our 4-alkylation chemistry, since there are 2 different probes available currently. That's the 3,4-methyl is a small one, but also this tetra-4-ethyl imidazole probe is a little bigger one. We tested the potential of this probe on the proton chloride exchange transporter from E. coli. And according to our experience, we are able to see modification of aromatic residues of this antiporter by the small probes. These are colored in the red, but also some modification happen using the bigger probe. But what's not surprising us, that using this big probe, we are only able to the amino acid chains surrounded by the detergent or by the lipids with the heavy probe. But the light probe also allow us to modify the aromatic amino acids right inside the channel. So if we are now planning to investigate the conformal rearrangement of the channel in the presence of different ket or anions, these small probes can already give us what's happening also inside of the channel. At the end of my presentation, I would like to thank you to the rest of my lab, which is located in the Bioset. It's a few kilometers outside the city, and mainly to Jan Fiala and Lukáš Pointy, making this chemistry available in bench4 setup, and Peter Mann for helping us and being so helpful preparing all these membrane proteins and understanding what's happening in the lipid barrier, and of course our friends from Israel supplying us with their samples, and Václav Matoušek and Peter Bayersdorf from Institute of Organic Chemistry and Biochemistry, helping us to design this for our kilo probes for our footprinting experiments, and of course to EPIC Consortium for running this webinar and supporting our labs. Thank you for your attention.
Thank you, Peter. So we are just about to open our last talk, and we are right on time. So let's pass to Argyris Politis.
Hi, everyone. So let me share my screen.
Yes, we can see your screen.
You see my screen? All right. Maximize it a bit. Okay, that should be in the maximum, right? Good. Okay, thanks so much for the opportunity to present my the work we're doing with my group in Manchester. So in my group, we are very much interested in a mechanistic understanding of membrane proteins, with particular focus on transporters and more lately on G protein coupled receptors. And the main technique that we're using for that is hydrogen deuterium exchange mass spectrometry. Quite a few speakers that have introduced this method and presented some great data on this technique. However, here I want to point a little bit about the method development that can facilitate studying these membrane proteins into the native-like environments. So, for instance, understanding the dynamics and the interactions within lipid-based environments, but also how we tackle with these methods for challenging targets. So, obviously, HDX mass spec is a very good tool to understand functional dynamics of membrane proteins, particularly after we know a lot of information at the moment about the static structures of proteins based on like the cryo-electron microscopy, but also more lately on using AlphaFold or the structure predictions using the artificial intelligence tools. So it's really the right timing to look into the functional dynamics of proteins in general, and this is HDX is one of the methods that can play a key role for these endeavors. Okay, sorry, I passed my slide. So membrane proteins, as you know, they're very challenging targets. So ideally, we want to study them within the native environment where they live and operate. So this is the lipid-based environment. This is a membrane, right? So that can protect the proteins from water, right? So the hydrophobic part of membrane proteins. So in that way, we deal with a rather heterogeneous environment, which can be quite challenging to incorporate into the normal workflows. But also, while they provide us a challenge, they provide us also opportunities to study. So these opportunities can be like how we can think about membrane proteins in association with the lipid environment, how the lipids can modulate membrane proteins, for instance, how they can interact, bind, but also like a shape, structure, function, and dynamics of membrane proteins. So we've been looking over the years, we have published extensively on a membrane protein field, particularly with HDX mass spectrometry, on transporters. For instance, we have been looking into sugar transporters. We're using Xylee, a protein that we express and purify in my group, and we as a model system in order to study sugar transporters, and this is because xylene is a homolog of GLUT1, which is a protein involved in quite a lot of pathologies, and is a human transporter. But we've also been studying ABC transporters, but also, as I said in my introduction, G protein cap receptors is one of the targets that are very sought after for many techniques, and that's something that we have been involved in, and I will show you some data later on. Before we started doing a lot of membrane proteins, we thought about we need some kind of a workflow, a method that can incorporate the lipid-based environment. And obviously, we're not the first coming into the game. There were other groups doing that in the past, but however, we sort of put it in more like a systematic workflow. And recently, like a few years back, we published a Nature Protocols paper, like describing how we deal with membrane proteins within the lipid-based environments. I'm not going to go into much details on that because previous speakers have described very well how HDX works, but I would like to point the attention on key steps that we incorporated into the HDX workflow while we're dealing with membrane proteins, especially membrane proteins where the lipids are involved into the experiments. So for instance, when we use nanodies or other native-like conditions. So we do need to remove lipids from the workflow. And the reason for that is that we don't want to clog the mass spec with the lipids, and also we don't want to have the lipids in the LC, which can like lower the quality of separation from liquid chromatography. So in order to do so, just after the quenching, right, and before the enzymatic digestion, we add a step where we remove lipids using different methods. One method that we've been using is the addition of zirconia beads. We can either do that offline or automatically. New instrumentations like water instrumentation that we're using, we can have the lipid filtration toolkit that we can actually do that online, like automatic manner. So we can actually remove lipids automatically, and then we can have them streamline more the process, be able to get more like clean data afterwards. But also one other thing that is very important for the membrane proteins, or for the HDX of membrane proteins and also proteins in general, is like the data analysis. As a lot of you know, that the data analysis with HDX is quite a laborious process. So usually in my group, for one day of experiments, we might need 1 week of data analysis. So for that reason, it's dealing on how we analyzing the data is an important aspect that we, especially in the beginning, I set up my group, I spent quite a lot of time doing with that, and we developed softwares. And for instance, one software we have is the Deuterus that can give us information about the data. And here I plot, for instance, which plots that we're using a lot for data analysis of HDX. We saw us the protection or the protections or maps that we can see the different peptides, and in that way we can have a quick output of what HDX mass spectrometry can give us. But I will move on a little bit of some examples that we have studied in my group over the last few years, and this one of the key proteins that we've been using a lot for developing our tools and methods, but also understanding mechanistically how membrane proteins work is the xylene transporter. As I said, this is a transporter which transports xylose from one side of the membrane to the other side of the membrane. In order to facilitate this transport, there is also some proton gradient involved. What is nice about this system that we use is because it's a well-behaving system in our hands. This is quite important for purified proteins and HDX, especially for people who are developing methodologies. But also there is a lot of information, structural information available. And this is also key in order to be able to interpret the data. For instance, if we have mapped the data, HDX data on three-dimensional structure, it's quite important that we have them. We have available structures that can see how they behave. So we have done experiments on this transporter by, for instance, by comparing the wild type with mutants. And one of the first experiments we did is to see whether we have any opening or closing of the conformation dynamics of transporters, and we compare the mutation. What we did there, we substituted a small residue glycine to bulky tryptophan at exactly the point that is preventing the transporter to close to the outward-facing state, and we compare that with wild-type conformation, and we saw in our results here that we can see the red bits on the top of the extracellular side and the blue bits on the bottom, the intracellular side, that we have an opening of transporter towards the outward-facing conformation. This is quite an important experiment because it showcases that clearly that in our hands, this methodology works, and we can probe conformation states of transporters. One addition here that the HDX obviously is a sample technique, right? So it averages out the different conformation. That's a limitation, but also in this case, we use it to our benefit because as you might know, the wild type conformations, they are like some of the transporters, they're freely moving. So as a reference state here, this conformation, it was more resembling the occluded state. So we use it in order to show the reference state as opposed to the open state of the mutant. But based on this experiment, we went on and we did some more interesting biological experiments that's related to a known intracellular gate of the transporter. We're sitting on here at the bottom of the transporter. By mutating, disrupting the interactions in this intracellular gate, we can show that mutants versus the wild type will have a shift of the conformation equilibrium towards the intracellular side, the inward-facing conformation, and that indicates that you know, like HDX can be used in order to study interesting biology. But further than that, we incorporated the transporter into like lipid nanodiscs. So we used like reconstituted into nanodiscs of physiologically relevant lipids, that's in bacteria, the PE, PG, and cardiolipids, and we compare that with PC-based lipids, which PC is not in bacteria, but however, we use that as a reference state. Again, when we do HDX mass spec experiments, we normally do differential experiments because that's how the information come about, and that's information depending on whether we have a mutant versus wild type or ligand and so on and so forth. In this case, we're changing the lipid composition and want to answer the question, what is the impact of specific lipids, PE lipids, into the conformation dynamics of transporters? For this, we use the Xylee transporter, our model system, but also we use another very well-studied transporter from the same family of transporter, the major facilitator superfamily, that's the LAG-Y. That's also well-known, very studied. And for these two different transporters, by just modifying the lipid environment within these proteins, we can see that we have a shift of the conformation equilibrium from the outward phase to the inward-facing conformation. You can see that with the red beads at the bottom. Obviously, that indicates us that there is some impact of lipids on conformation dynamics, and wanted further to confirm that by doing some MD simulation with our collaborators in the States, in Illinois, Matt Tatskosit group. We carry out simulations, exactly the same lipid composition as the experiments, and we can show here that we have like interactions of PE lipids with the intracellular gate that we have identified previously on our initial experiments. And this has prompted us to go on and do like mutations on this interaction side, and then go back to HDX mass spectrometry and show that actually if we do the delta P versus PC lipids in the mutant, where we have the interactions of lipids to the protein, that we can abolish the effect we have seen before. That tells us that we have a definite protein-lipid interactions of the proteins. So, however, that was one part of our studies, but I went on to understand the mechanistically how the protonation and a substrate and inhibitor binding on xylee transport, and we carry out quite a lot of experiments without inhibitor or substrates, but also mutants, which mimicking the protonation site. So we did that by substituting negatively charged mutants residues to uncharged residues. So in that way, we mimic the protonations. So I don't want to spend too much time on that, but I would like to point you to the key findings of our research that we published a couple of years ago, that with HDX mass spectrometry, we see here what we saw, that single mutation has different effects than double mutation. So we can have like a specific like mutations that can show that we have differences in conformation dynamics here. So double mutant can shift the conformation equilibrium into inward-facing conformation, while a single one, they have like a limited impact on the conformation dynamics. We can go on further and see how what is the specific mutant that plays a role into this driving these structural changes. And here we can show that the D27, when we compare the double versus the single mutant, they have like clear changes towards the inward-facing conformation, where when we manipulate the 206 mutant, there's no such changes. What was more interesting in this study was trying to see whether we have any differences between substrates and inhibitors. So there are crystal structures of the xylee with substrates and inhibitor binding that shows not major differences, like pretty much the same binding site, the same conformation. However, based on what substrate and inhibitor does to the protein, this could be the full truth of the story. So that's why we wanted to investigate that with HDX mass spectrometry. And here you can see that when we compare the conformational dynamics between the glucose binding with the inhibitor and the xylose binding to xylee, we have very much different conformational fingerprints. At the top, we see like obviously something that we expected, an outward-facing conformation locked when the inhibitor binds to the protein, meaning that precludes the transporter to move the biomolecule to the other side of the membrane. But when we have the substrate and we have more transient open state conformation, that indicates us that it was some intermediate state that potentially high resolution structural biology methods couldn't capture because of some of the limitations might have at the time of sample requirements. We will be able to corroborate these results with an MD simulation, where we carry out experiments, the similar conditions on the protonated states with and without substrate inhibitor. We can see here that we have way more flexibility when the xylose substrate bound as opposed to glucose. And obviously all these, we put it together in a substrate dependent energy landscape, when we can propose here the sequence of events and what binds first in terms of protonation, where the protonation happening, which residues, what comes first, and then the substrates come into the game. Obviously, the lipids have a clear effect that shifts the conformation equilibrium into the inward-facing state, and this is complements the cycle of membrane proteins. So here I want to present a little bit some work that we're doing in integrative workflow to unravel conformation population of proteins. And what we do here, we're using like HDX experiments, but also molecular simulations in order as a source data to try to have to quantify the conformation populations of transporters. One of the problems we have in structural biology is that some cases we know that there is one state or another, but we don't know how much of the state is there, right? We don't know that if it's 100-0, or if it's 70-30 or 60-40. That research we're doing in my group, we're trying to address exactly this question. So what we developed, like combined experimental computational method, when what we do is like to perform reweighting. So in a way that we can start with initial guess, and then using a bias, a correction factor, we can reweight so we can match experiment with simulations. at the end of the day, that give us the possibility to quantify the populations of our proteins. And obviously this can be applied on different type of techniques, or different type of, cases, like mutations, lipid environment, changes in lipid environment, ligand binding, but also allosteric modulations and regulations. We have tried that to mutants, but also like different lipid environments. You can see that we can have a different type of populations that we can monitor through our experiments and the subsequent reweighting simulations we carry out. We have also like implemented that in different substrates, ligands like substrate xylose or xylene, also the glucose, and different non-inhibitors of the GLUT1 transporter, and we can have like a functional population of the different outward and inward conformation of transporters here. Obviously, this is something that we're working more in the future, is that how we can actually monitor, how we'll be able to capture intermediate states, and something that's quite an open question in structural biology. But for the very last minutes of my talk, I want to talk about an exciting frontier in our research, and that's studying G protein coupled receptors. We studying a series of GPCRs, but one of the key GPCR we're focusing on to develop methods is the beta-1 adrenergic receptor. And this is like a well-studied again system. So I don't want to get too much into detail, but I want to say that we have spent quite a lot of time to optimizing conditions on a GPCR, a very difficult, unstable target to reach around 95% of the overall sequence coverage. And we went on to study how agonists, but partial agonists will be able to affect the conformational dynamics. And here we can see, like, when we have different agonists, we have like a clear vision of the binding site with a transmembrane helix 5. We have, in all cases, the binding site, but also we have like a conformational closing that's happening at the in the intracellular site. When we have agonists, we have completely different fingerprint. We see a conformational activation in the intracellular site that comes also with the binding of the transmembrane helix 5. And partial agonist is sitting in between those two. And in that case, we want to point attention that actually HTX mass spec can really be used as a tool to probe like compounds, what's the effect on GPCR, something very powerful. Something we're investigating more, and that I will close with that, is about the ICL1. Is something that we have seen is a loop on this GPCR that can give either protection or deprotection on antagonists or agonists respectively. And we've been working on studying a bit more and see the impact it has in dynamics, but also in functional dynamics of the proteins. And here we can have designed experiments. We have like a reduced efficacy of the different like agonists, or the isoprenaline, for instance. And here we can see that we have like a different levels of impact on the ICL1, and we want to take this further as a more biological insight in our research. But anyway, I don't want to, I think I ran out of time, and I want to acknowledge all the people who did the work, the collaborators, but also the funders and yourself for attention, and happy to take any questions.
Thank you for your great talk. Yeah, and we are right on time, so there is enough time for questions. So have a look in Q&A. There is a question for Gian Langer, if it is still available. So the question is: cyclic IMC shows better sensitivity for deuterium uptake and detection of peptides originated from transmembrane domain. I might point out to IMS for the detection of low abundance species, but did you observe different deuterium incorporation for the same peptide, non-cyclic versus cyclic? And
yeah,
if I may, I would actually double follow up on the question. Obviously, what you saw, the different uptakes go beyond between the two instruments, go beyond the differences in sensitivity or resolution. So do you have some explanation for it, like a different back exchange in the two instruments? Or
so on paper, the two instruments had the same front end, and apart from the if we exclude gas phase back exchange, they would have the same, they should display the same back exchange rates. I actually showed a couple of peptides on one of the slides, and you could go back to that, where we have the same, we have the identical peptide where we saw just a higher uptake. So this may be because we can still track particular deuterated peaks in the cyclic data, which we can't see anymore which disappear into the noise in the synapse data. So that's my interpretation of what we see there. So in terms of back exchange, you could argue if you have a longer gas phase back exchange, that would actually work against the cyclic, right? So yeah, but still, I mean, it wasn't the same front-end instrument, so I can't really say if that was a non-issue or not, but we made sure that the capillary length and everything were identical. So it's kind of may have been a bit colder in one lab than the other. I don't know. I don't think so. I mean, we had it as controlled as we could. Yeah.
The fact that the cyclic has a better mobility resolution, could that play a role?
Yeah, absolutely, because you essentially you're removing a lot of otherwise overlapping peaks, so that really helps a lot. And I mean, if you want to let me reshare real quick, I can just show one slide here.
Sure. I'll stop sharing. So it's all yours.
Yeah, just going to show this real quick. I mean, if you can see that here, these are identical peptides. The full line is my cursor is now moving a bit slowly. The dot. Oh, did I just? Can you still hear me?
Yes.
Okay, so I just got a notification that Zoom quit unexpectedly. Didn't have that before. Anyway, so what I wanted to say is that the dotted line is synapse data, and the full line is cyclic data. And this is the same peptides that you can see here. Okay, except for this one, which we only had on the cyclic. But you can see that our uptake rates were just higher across the board. Yeah, so that's our interpretation for these data, and this has all been uploaded to Pride. So and it's published. So if you want to dig in, we're also looking at it. We acquire more data sets, so let's see how it plays out. So.
Interesting, thank you.
Yeah.
And I might have one question for you actually, or it was very interesting to see the P116 as kind of like a molecular enriching system for the lipids. Do you have some? Is there any structural information? What sort of lipids being enriched? Is it like, let's say, phosphocholine or phospholipids?
Yeah. Okay, hold on.
The fats are being left in the plasma, or do you know the structural features needed for the enrichment?
Yeah, I just rejoined. I'm sorry.
Okay.
I think there is an update running in the background on my PC. So what I wanted to show here is, so we actually took a close look at which type of lipids are picked up. And this is essentially the entire lipidome with all these different lipid classes that we detected. In total, more than 500 different species. So we could very clearly see that PCs, so phosphatidylcholines and sphingomyelins and cholesterol esters were picked up selectively from the bulk serum lipids that were present overall. So in the serum mainly, you're going to see TGs.
Yes, okay.
And these were hardly picked up at all. Like we had only like trace amounts of that there. And the dominant species were actually PCs in the sense that are not exactly representing the dominant or the bulk of the lipidome in the serum. So we see specificity, and we have no idea how yet.
Okay.
So that's what we'll be looking at next, actually, mechanistically.
So there are no more questions in the chat, but anybody would like to ask a question? Anybody raise his hand or just start speaking? Well, if not, then we are coming to an end. So on behalf of all the organizers, I would like to thank everybody for attending, and also to everybody who supported the EPIC during the last 4 years. It was really great, and thank you very much.
Yeah, I just would like to say thanks to all the speakers for their time today and their participation. And yeah, with that, then I think we can end the webinar. So thank you, everybody, and I hope you have a nice evening.
Thank you. Good night, everybody.
Bye.
Thank you. Good night.
Thank you. Bye.
Thank you. Bye.